Morning, ladies and gentlemen. This is Serena, your today's host for the conference. I'm a program manager at Platinum Global Solutions, and it's a great pleasure for me to welcome you all to the sixth annual Global Drug Discovery Summit 2021. Thank you all for joining this virtual meeting, and I hope every one of you enjoying their best health. So, whether you are attending this event for the first time or you have been a part of our top notch conferences before, I'm sure that your experience at this drug discovery e conference will be worthwhile and productive. So, our conferences have a reputation among delegates and suppliers as a must attend event. I hope that the highly focused business development opportunities through the cutting edge conference program and the numerous networking opportunities will all contribute to a successful and enjoyable event for you. So now I would like to warmly welcome our key presenter for today's conference. Our first presenter will be Guillaume Plain. He's Marketing and Development Manager from Merck. He will be sharing his expertise on current trends in monoclonal antibodies manufacturing. Then we have Antonio Gomes. He is a Director Alliance Management at the Janssen. He will be presenting on new strategies in the drug discovery industry. Then we have Revital Ratzenbeck, CEO and Chairwoman from Forby Pharma. Her thoughts will be on fast development of a CXCR4 antagonist to fight against COVID-19 pandemic. And then we have Fred Jacobs. He is a CEO from TYG Oncology. He will be speaking on the world's most dangerous hormone. And then we will be having a 15 minutes short break for your coffees. And then we will be having with us is Musa Yodium. He is the president, CSO, and professor emeritus from Yodium Pharmaceuticals. His topic is why multi-target drug for neurodegenerative disorders. And then we will be having with us is Kiel Stenberg from Exenta. He will be covering accelerating drug discovery by AI and repositioning. Our seventh presenter will be Raul Insa, founder and CEO from Som Biotech. He will be exploring AI solutions in drug discovery applied to orphan indications. Then we have Dror Heratz, CEO at BPL Therapeutics. He will be sharing his expertise on transformative gene therapy with the potential to change the treatment paradigm for solid tumors. Then we have Eric Oliova, President and CEO from PDC Line Pharma. He will share his expertise on new class of cancer vaccines based on an off-the-shelf antigen-presenting cell line, PDC line. And then we will be, that's it for the speaker lineup. I hope that we will learn new ideas from each other, which could help us to improve the work in drug discovery industry. So our first presenter is Guillaume Plain. He is with us, and let's welcome Guillaume. Hi, Guillaume, can you hear me?
Yes, can you hear me?
Yeah, I can hear you. Can you please start your video?
Sure.
Great, I can see you now. How are you?
I'm fine. How are you?
That's good. I'm good. Thank you so much for asking. So, ladies and gentlemen, this is Guillaume playing with us. He's working as a marketing and development manager at Merck, and he's a biochemist led by an entrepreneurial spirit, proactive, mobile, and result oriented. Listed as a biotechnology biomanufacturing expert at BPI, and recently graduated in law. Currently preparing a next professional step. His specialities are in biomanufacturing, biochemistry, marketing strategy, and business law. So, Guillaume, thank you so much for being here. We are honored to have you as a keynote speaker. And you can start your presentation now by sharing your screen. And okay, and I'll ask the audience that if they will be having any questions, they can ask in the QA session or in the chat session, okay?
Sure.
Okay, so Guillaume, the stage is all yours now. Good luck.
Thank you. So today I'd like to share some consideration around the biomanufacturing of new drugs. Discovering a new drug, especially for unmet medical needs, is something, of course, very great and very important for for the future, but being able to manufacture, to produce this drug is something critical in order to address the market. And this is something that has to be considered at the very beginning of the drug development. If the discovery of a new drug is very early, and if it's a biological drug, it would be a sequence of DNA, then most of you know that it takes a long time to reach the IND filing step, which allows people to go through the clinical studies through phase 1, 2, or 3, and then a market approval. This full process can take from 8 to 15 years, depending on the complexity of the drug and depending on the complexity of the disease. And as you can see on the next slide, the costs for the drug development is huge at the beginning, and at this time, people don't make money at all. Thanks to their new drug development, they need to wait for commercial approval in order to get revenues from sales, which, depending on the IP linked to the drug, is more or less, let's say, 10 to 15 years. And then you have what we call the patent cliff, where the drug is now public and everybody can copy the drug. That's the case for generics, but can be the case for biological drugs as well. And that's what we call biosimilar drugs. As you can see, the cost is huge at the beginning until market approval, and it is reduced, only made of marketing and cost of manufacturing after market approval. But at the very beginning, the costs are made of clinical studies and process development and clinical batches manufacturing, which can be huge and has to be considered, as I told you, very early in the process of developing a new drug. So what's the current pipeline? That's not exactly the current pipeline. It's a little bit older. It's 1 or 2 years old. We currently have more than 50,000 products on the market. What I call a product is a modality on one market from one company, which can be a little bit complex, but if a drug is commercialized by 2 different companies, then we consider this as 2 drugs in this study. So as you can see, for the conventional products, the products which are mostly based on chemistry from the, I would say, traditional pharma industry, most of the products are already on the market. We have some products at preclinical stage, some products at clinical stage, but compared to what we have for biotech products, where most of the products are at preclinical stage and let's say 1/3 on clinical stage, and only, let's say, perhaps 15 or 20% of the products on the market, this biotech market is still under development. It's a market in the making. It is not yet mature, and we can learn from what happened on the pharma market in the past in order to understand and influence what's going to be the biotech market in the future. So what's the challenge for the biotech drug development today? This is the molecule of the aspirin. You know, aspirin, this very old drug, which has been discovered, I think, 1.5 centuries ago, and which is still used in the pharma industry. But of course, it's now used. It can be produced by anyone in the world. There is no more IP under this product. It was made of 21 atoms, so it's complex for chemistry, but compared to what you can see on the right side of the slide, a monoclonal antibody, which is made of more than 25,000 atoms. It's much more complex, and you can compare these molecules as you can compare boats on the sea. And I would not say that this boat on the right could be called the Titanic. Most of the drug makers hope that it will not be a Titanic, but it's very complex to drive, very complex to develop, as a large boat like the one on the slide is very complex to drive. So this has to be considered for the biotech industry today. So the current pipeline of the biotech product, as I told you, is made of something like 15,000 different projects or products, and most of them are the preclinical stage. We consider that this product, this market, is a 250 billion market. It's a huge market, but the diversity of the market has to be considered. Most of, or the largest part of the market is based on monoclonal antibodies, and we currently have hundreds of these products on the market today. So it's really the gold standard of the biotech market today. But as you can see, some recombinant products are a large part of the market. Otherwise, the market is split in many different categories of products, from gene-modified cell therapies, what we call, most of them are called CAR T cells, gene therapies, DNA, RNA. Of course, RNA is now a very promising modality following the market approval for the mRNA vaccines against the COVID-19. But the monoclonal antibodies is still the main part of the market, cell therapies, and we also have antibody drug conjugates. For all of these categories of products, for all of these modalities, the way the manufacturing of the drug is performed is different and has to be considered for the next steps. So the current pipeline and the current by modalities is very different from one category to another. For the recombinant products, which could be insulin, erythropoietin, EPO, which could be factor V, etc. These products are mostly already on the market. They were developed at the very beginning of the biotech industry development, thanks to companies like Amgen, Chiron, Genentech, Biogen, and we could almost consider these products as a mature market today, which is not the case of the monoclonal antibodies, because most of them are still under preclinical development. Some are on the market. As I told you, hundreds of those are on the market, and many are under clinical development. If we compare this to DNA and RNA, you can see that the number of the projects is much less important, but most of the projects are still on preclinical development. But this modality is still a promise. It's really a promise following, as I told you, the market approval for the mRNA vaccines from Moderna and Pfizer BioNTech against the COVID-19 disease. And another modality, which is still at the beginning of its development, but very promising for the future, is gene-modified cell therapies. We currently have 2 or 3 of these gene therapies or cell therapies, on the market from Novartis, based on the CAR T cells. but many are under clinical development and under preclinical development. And that's really a promise in order to fight the cancers. As I told you, recombinant products are still in the mature phase of the development. Many of them are already on the market. And what we can put together is the stage of the development of a given modality and the fact that this modality is templated for the manufacturing. As I told you, the manufacturing has to be considered at the very beginning of the drug development. And if there is a template, it's much easier for the drug makers to envision the future. For the recombinant products, the templates are here. We have bioreactors for microbial growth during the upstream stage of the manufacturing. And we have many systems for the harvesting and for the purification based on chromatography and filtration. And they can be scaled at the industrial production scale, which is not necessarily the case for new products. For the monoclonal antibodies, we have a template, or we have templates for the manufacturing. It can be improved, and many developments are currently performed by the industry in order to reduce the costs, in order to increase the volumes and the agility in the facilities that we use for the manufacturing. But this is still something that I would say is easy to manufacture. Not very easy, but easy to manufacture, which is not the case for gene-modified cell therapies, where we don't have any templates. We now have templates for RNA manufacturing, thanks to, as I told you, the market approval of the mRNA vaccines. And given the number of tech transfer which have been performed by Moderna and by Pfizer, BioNTech, for this modality, we could consider that the production of this messenger RNA is now under control for the whole industry. If we consider the monoclonal antibodies, which is, as I told you, the gold standard for the new drug manufacturing, 2 or 3 years ago, the main part of the market was made of 10 molecules, which were even more than blockbusters. Herceptin, Avastin, this molecule generated billions of revenues for the drug makers, but they are now, or they will be very soon, near the patent cliff, meaning that anyone in the world could copy these drugs and provide them to the patients. So from 2018 to 2024, the part of the market segment of these 10 main molecules will decrease from almost 11 to 6. And the part of new products will increase from 5 to 16, meaning that we consider that many new products based on monoclonal antibodies will reach the market and get a market approval, most of them being developed in order to fight against the cancer. But another part of the market is made of biosimilar drugs. These molecules, these 10 molecules, can be copied as biosimilar drugs, and the decrease here from 11 to 6 will trigger an increase in the biosimilar drug development from 1 to several billion revenues in the future. So there will be a growth of the biosimilar drugs. The top 10 drugs will become like very will decrease in terms of market size, and new stars, new amazing drugs will reach the market within the next 5 years. And this allows us to define trends in the biomanufacturing industry. And the main trend, and that's a current trend, and I can tell you that we feel it in the biomanufacturing industry, is the cost pressure. And the biosimilar drug development triggers a cost pressure, which is huge. People developing biosimilar drugs, what the only competitive advantage that they can get thanks to their biosimilar drug development is based on the price of the molecule, mostly on the price. And the way they can decrease the price is a decrease on the cost of goods, the cost of manufacturing. And this decrease in the cost of manufacturing is possible if you increase the yield or the titer when you make the manufacturing. And that's the difficulty for the manufacturing industry. Decrease the cost of goods. Other trends based on market growth, and that's a good news, of course. Emerging markets welcome biological drugs, which was not the case 15 years ago. And this triggers new trends based on regional manufacturing. You don't have one large facility for the global market. You now have facilities for the regional markets, which can be used for several drug manufacturing thanks to single-use systems. Uncertainty and new product classes is another, are two other trends of the market development and has to be considered. If we consider the biosimilar drug development, as I told you, the cost pressure and the market growth are the main threats which can be considered. And this is something which will deeply impact the manufacturing industry for the future, because by this year, 50 billion per year in biological cells will be off patent. I told you Herceptin, Avastin, many other drugs which were amazing blockbusters for the drug makers are going to be IP free. So many of biosimilar drugs are currently under development, and the competition on the market will be completely different in the next 5 years compared to what it was in the past 5 years. That's a huge breakthrough in the way we produce these drugs, and this will impact the production costs of the drugs. Before that, we did not really care about cost of goods. We now really care about cost of goods, and we always try to improve the yield of manufacturing in order to address these trends on the market. The biosimilar drug development is mostly, as I told you, made of the switch from the 10 main manufacturing drugs to 6 billion per year, increasing the biosimilar part, and mostly for these molecules, Rituxan, Avastin, Herceptin, Remicade, and Humira. The change in the volume is huge, as you can see. We anticipate almost more than 50% impact in the decrease of the drug manufacturing for the originator, which will be replaced by biosimilar drug. Another trend on the market is uncertainty and new class of products. And for this, adaptability is really important. And this can be obtained thanks to single-use manufacturing. Adaptability, you can see on the left part an old stainless steel facility, which is not very agile, or which does not allow us to be very agile for the manufacturing. On the right, you can see a single-use manufacturing. It's a 200 liters bioreactor manufacturing with a harvesting system. And this allows us to use the same suites of manufacturing for the production of different drugs, which was not the case before. Thanks to single use, we will increase the flexibility in the facility. Last slide. In order to illustrate the trends on the biomanufacturing market, we have the cost pressure, the uncertainty, growth in the market, and class of new products, and the BPOG, which is an organization, an association of drug makers, going to think together on the future of biomanufacturing, defined the challenges for the future on the cost, a decrease by 80% in the manufacturing costs, mostly based on the competition. A flexibility, and this will be allowed thanks to single-use manufacturing, a decrease in 90% of the changeover. An increase in the speed, not only the speed from DNA to IND submission, not only the speed from IND submission to market approval, but also the speed from an order to the delivery of a batch of a drug. The speed should be increased by 80% for the lead time and 70% for the build time. And quality and robustness will have to be improved in order to define the facility of tomorrow, which will be based on process technologies. The facility will be automated, modular, mobile, of course, an inline monitoring and real-time release will be allowed in this facility, with the management of the knowledge will be improved, and the management of the supply chain will have to be improved as well in order to reach these goals of the decrease of costs, the increase of quality and speed, and flexibility. We at Merck have a CDMO activity for mAb manufacturing. We have 3 different manufacturing facilities, one in France near Bordeaux in Martillac, one other in Shanghai, and one other in North America, in Burlington, Massachusetts. And we help drug makers develop their process for monoclonal antibodies and reach their IND submission and reach their market approval, thanks to support for batches manufacturing through clinical and commercial development. And that's the end of the presentation. If you have any question, of course, I would be happy to answer the questions. Can you hear me?
Yeah, I can hear you. Hi, Kiamat. Thank you so much for your wonderful presentation. It was really very interesting, and thank you so much for being here. And I'll ask the audience that if they will be having any question, they can ask you in the QA session or in the chat session. Okay. Thank you so much for your wonderful presentation.
Thank you.
Have a great day. Stay safe. Bye. Okay, so ladies and gentlemen, hope you have enjoyed the presentation of Biomanufacturing because it was really very interesting. So now our next presenter is Antonio Gomes. He is with us. And hi, Antonio, can you hear me?
Yes.
Okay, great.
Yes, I can hear you. Can you hear me as well?
Yeah, I can hear you and I can see you as well. So, ladies and gentlemen, this is Antonio Gomes. He is working as an Associate Director Alliance at the Janssen. And Antonio Gomes has almost 25 years of experience in the pharma industry. Right before getting a PhD in synthetic organic chemistry, he joined the Janssen R&D group in Toledo as a medicinal chemist in 1996. And starting in late 2013, his role has been integrated into the position of new venture lead in Spain for London Johnson & Johnson Innovation Center. And since 2017, as an ASAP certified alliance manager, he's also involved in the managing external alliances for Jensen at a global level. So let's welcome Antonio. So hi, Antonio. How are you?
Fine, fine. Let me, I guess, share my screen.
Yeah, sure. So you can start your presentation. I can see your screen.
Right, let me go to
Full screen, yeah.
Okay, that's it.
Okay, so good luck. Stage is all yours.
Okay, so thank you very much, Sarina and Platinum Global Solutions for giving me the opportunity. What I want to do in my presentation is really to give an overview of first what we are doing in the pharma industry, why we are doing it, or well, more than in the pharma industry, I would say in the drug discovery process in general. So it will be what we are doing, why we are doing it, and how we are innovating on, or how we are creating new solutions. So it will be, as I said, just an overview. So there will be in this summit, there will be very good speeches about some of the particular topics. So I think you will have much more information on them. And the idea is to have this high level view on all of this. So let me start by, well, this is like a corporate slide that I took from a slide deck, because I think it summarizes very well what we do and how we do it. So here there are three very important words: heart, science, and ingenuity. Along the presentation, you will see science and ingenuity mentioned once and over again. Instead of science, I will be using a synonym, which is knowledge. Instead of ingenuity, I will use innovation, but it's basically the same. And then, yeah, the other very important part of this slide is changing the trajectory of health for humanity. So what it means is that what we are dealing with here is health. And if I go to the next slide, which is also like a corporate slide, here you see I will start in this square and then I will go counter anti-clockwise. And first you see that in Johnson & Johnson, we are made up of three business segments. The first one is pharma, and this is what is referred to as Janssen. Then there is another one on medical devices and a third one on consumer. But the common denominator of all three sectors, of the three pillars, as we like to call them, is health. So in all cases, we are dealing with human health. If you go to all these squares at the bottom, basically, if you want to summarize them, it means that we are a very large company. We are very big. We have a lot of products. We are present by creating new solutions for patients, or for, in the case of consumers, for consumers worldwide. So let me start on the first part, which is on what we are trying to do, because sometimes we know, of course, but sometimes it's worthwhile to go and reflect a little bit on what we are doing. So we are developing drugs, right? So this is a drug discovery summit. We want to discover new drugs. And what I did is take the definition from Encyclopedia Britannica. So I didn't really, I tried to go to sort of golden standard. So you see that drug is chemical substance that affects the functioning of living things. So it's something that we take and that changes the way that we work, that our bodies work. And then disease, what is disease? Because we want to apply drugs to disease, so to change from a disease to a healthy state. And here I think the important part of the definition is harmful deviation from the normal structural or functional state. So of course, what is the normal state? And this can be very, very controversial, but then it focuses a little bit more because it says associated with certain signs and symptoms. So we have some signs. There are some things, there is something in our body that is telling us this is not working well. You need to do something, and it's harmful. So this is the other important part of this definition. So what we are trying to do is no less and no more than this. So we are sick, we are feeling bad, and then we want to be like this guy on the right side. We want to feel okay. And we want to do this by taking something, by taking this drug that was present in the definition in the previous slide. So we take the drugs and we become healthy. But yeah, this is what we see at the, let's say, at the phenotypic or at the clinical level. But we all know, because yeah, we are in the industry, but I think in the general public, everybody knows that there is something going on at another level. So we change from this state to that state on the phenotypic or clinical level by taking something, but there is something behind, which we call mechanism of action, that is making these drugs work in the, yeah, in another level. And which one is this other level? So at the beginning, it was magic, right? So you took something by whatever, by some magical spells, by some magical properties of whatever we were taking, then at that level, there was a change was being produced at the magical level, and that level was translated at the phenotypic level in healing the disease. And I have to say that probably this magic was was working pretty well, especially because I think the data that I read, the last data that I read, is that 80% of the time that you are feeling bad, just by doing nothing or by doing some magic spell, you will heal. So yeah, 80% success rate, that's not bad. Now, of course, we are not in that paradigm. Our current paradigm is that this level is not magical, it's biochemical. So we have at the biochemical level within our bodies some of substance, the active ingredient in this pill. So the active drug is interacting typically with a protein, and by interacting with that protein, something is changing within our body, and that change within our body that is being triggered by the action of the compound of the drug, that in turn, that makes this change at the clinical or phenotypic level. So yeah, might be that we have made like a very big leap forward between the guy on the left and the cartoon on the right. But yeah, if we go to this quotation that I really like from A.C. Clarke, any sufficiently advanced technology is really very different to tell from magic. So in fact, we now say, in the everyday speech, yeah, just do your magic. So this is what we want to have when we are feeling sick. We want to take something, we want it to make its magic, and then that's all. So it might seem that it's basically the same thing, but there is something very, very different, and it's really the capacity to build on top of what we know of the remedies that we have, to try to build on top of that new remedies. So if we think about that, from magical spells, yeah, you can have a magical spell to cure something, but then if you try to build on top of that to create another magical spell to cure, let's say, cancer, I don't think you will be very successful. Whereas in the current paradigm, which is based on science, we are, in fact, we are very hard working on the scientific knowledge that we have to try to create new solutions. And I think we are now in a stage where more than 50% of all tumors are cured. And yeah, we are working, let's say, there is still a huge medical need, a huge unmet medical need, but we are working on the rest. So if we want to summarize this difference, that's knowledge, and innovation. So knowledge, and that's the science. We are getting all this knowledge, and by having all this, and I really like this cartoon, by having all this knowledge inside, we can create innovation. That means that we can create new drugs, we can create new solutions for patients, which in turn we are going to feed into the knowledge, because this innovation is going to generate new data that are going to belong to the body of scientific literature as we know it today, and from that scientific literature that will enable other people that are, again, quoting very important people, standing on the shoulders of giants, building on science, they will create new innovation. So, and this is what we've been doing since basically the start. What you see there, that's the Ebers papyrus, which is one of the first examples of medical literature. It probably, I think it's from the 1500s before Christ, but it probably is a compilation of knowledge which was acquired much earlier. This is, and this is no hazard, no chance that it's Egyptian civilization. Why? Because the Egyptian civilization, sure, they had this magical component to health. They were a very religious people, ancient Egyptians, so they were doing lots of spells, the text of pyramids and things like that. But they also started to get a lot of knowledge. Where did that knowledge come from? They had two main sources. One of them was mummification. You know that they mummified the dead bodies. So by mummifying, they acquired a huge, spectacular knowledge of human anatomy. And then the second one was chemicals. So they knew not only chemicals from plants, which what we would in general today call organic components, like Guillaume was mentioning before, aspirin. Aspirin was not known to the Egyptians, of course, but what was already known was willow bark, which is salicylic acid. It's an active ingredient, and which is not as active as aspirin, but it already has some activity, and it was already known to them. So I'm not sure if it is in the Ebers papyrus, but probably it is. On top of that, they acquired a lot of knowledge of inorganic compounds. And they had in the desert, they had these mines, they started to use inorganic compounds for the process of mummification. And then very soon they realized that they also had some curative properties. So in fact, one of the theories of the origin of the name chemistry is that it actually derives from the name ancient Egyptians gave to their country, which was Hem. Hem meaning the black land, because it was the land that was enriched every year by the mud that was carried over by the Nile. So that's the Ebers papyrus, which starts to have some remedies for common diseases. And this is the first one of the first examples where you have knowledge, compilation of knowledge. On top of the knowledge, we've been building innovation. That innovation has created more knowledge, and this is where we have arrived today. That's medical science today. And if I start here in the second row, where are we now? So HIV, which seemed like really a fatal condition when it started not that long ago, now it's near normal life expectancy. That's really a big deal. Then we have a lot on targeted therapies, personalized solutions, robotics, digital surgery. So not only in the field of drugs, but in medicine in general, we have operations are now carried out with robotics. We are using more and more also data, analysis of data, to inform the treatments that we are going to apply. And we are also using long-acting treatments in general, and this I left for the end of this slide because I think it's the most important data. 60% increase in life expectancy since the beginning of modern medicine. That's since the beginning of the Industrial Revolution. So I won't say that this is all because of the pharmaceutical industry. I would like to say that, but I have to acknowledge that there have been huge changes in hygiene, for instance, in the use of sanitation. Also in knowledge, we now know that water and washing ourselves and taking showers is very good, which seems obvious to us, but 2 or 3 centuries ago, doctors were prescribing not using water at all because they thought it was harmful. So yeah, that's something. But of course, the modern medicine has a lot to do with all of this. And yeah, why have we been so successful up to now? So as I said before, we started with these guys, and yeah, I don't want to paint them in a bad light. I think these guys, they already possessed a lot of knowledge, which came from using herbs, using natural remedies. Even if they didn't have the knowledge on why they were acting, they knew that they were doing some magic at another level. They just were applying that they were thinking that it was a different type of magic. So on top of that, we have evolved a lot. So we have, of course, the scientific revolution of the 17th century. And there you have the advances in anatomy. That's probably since the time of the ancient Egyptians, looking at the human body, the dead body was like a taboo, religious and also cultural taboo. And this is the time when humankind overcame this taboo. And then, yeah, just as an example, another guy, this is of course Louis Pasteur, and he's the father of microbiology, understanding infectious diseases. And we cannot forget that infectious diseases were the main cause of death and disease in humankind until modern times. Yeah, as we all know, some of the diseases that are affecting most humankind today, like cancer, like neurodegeneration, they are in fact there because we overcame the burden of infectious diseases. Before, most people were dying rather young of an infection, and yeah, basically these new diseases, they didn't have the time to show up, and this is where we are now. So again, the same picture as before. We are now in this paradigm of modern medicine, of molecular medicine, looking at the molecular target and acting on that level, and knowing that we are acting on that level, and knowing how we are acting on that level. So can we be happy about this? Are we at the end of the story? Yeah, of course. You know the answer. The answer is not. And I just said that we have overcome the burden of infectious disease, but only to find new diseases that are more related to aging. So there are still lots and lots of unmet medical needs, and we are working hard in many different areas to try to win that battle as well. So let me start from the more, let's say, general to the more scientific and probably more fun for most of us, at least for me. But these are also important. So new approaches. This new approach that I'm here exemplifying with Johnson & Johnson model. This is the new approach of open innovation. So in the past, pharma industries, we were basically relying mostly on our internal R&D. And at some point in time, we realized, which is very logic, but for some reason it took us some time, we realized that most of the knowledge, most of the innovation that was taking place outside of our pharma companies. So this is the model that we created. We already had this business development branch, which was tapping into late stage opportunities that were close to closer, I would say, already with a proof of concept in humans. And where we were basically providing our financial muscle and also our know-how on clinical trials. And then what we started in the last decade, in the middle of the last decade, was all this effort with the innovation centers, basically scientific experts which know the needs of the company, the strategy of the company, but which are also very that are also scientifically very knowledgeable and which know where to find good science, which can have, which is promising to bring, of course, a new solution to the patients, a new drug to the market. Then we also have JJDC, which is the venture capital branch of the company, which is financing, which is funding those interesting opportunities that have promising science. And we also have the J Labs model. The J Labs is a series of incubators, company incubators, where we basically host different companies which, as I said before, which we believe they have a true potential to deliver new drugs or new solutions to patients. Again, and you see that from a few slides ago on, I've been putting this at the bottom of each slide: knowledge, innovation, the feedback between knowledge and innovation, which is making us move forward in medicine. So another approach, another very interesting approach, disease interception. So if you remember from my first slide, I defined disease, well, Encyclopedia Britannica defined disease as having some symptoms. And this is the way we have been dealing with it really always. So if you have symptoms, you are sick, and then you try to heal. If you don't have symptoms, you don't do anything. Why should you? You don't have symptoms, you are feeling okay. But then by knowing more about medicine, we have realized that there are many diseases that are starting to develop much earlier than we have symptoms. And by the time they really show up, many times it's very late. And we all know examples. We know, for instance, cancer. Cancer, most, or I would say probably almost every cancer is curable if detected in early stages. Mostly by surgery, but there are other means as well. But in many cases, when they are detected, they are already spread around the body, and they are much more difficult to treat. But it's not only cancer, it's the same story with Alzheimer's, which you can see on the right. When we get symptoms of Alzheimer's, it's been developing for at least 1 decade. And it's at least with the current level of treatments that we have, it's already too late. But also with diabetes, which leads to high pressure, they are silent killers, which you are not aware of, but that they are creating the conditions for other more serious diseases, which will be very difficult to treat when you feel the symptoms. So again, this is not new, liquid biopsy. So we have been taking blood tests to know our blood pressure. Blood tests, and we are having our blood pressure measured. What we are trying to do now is with this liquid biopsy and with the help of these omics technologies, which is basically taking advantage of all the developments in the field of technology in the current world, trying to identify new biomarkers that are easy to identify from, as I said, from a blood test, and which tell us that you are at risk or that some disease is starting to develop. That way you can treat that disease very early on before you have the full-blown disease, and it will be much earlier to treat. So as I promised, I'm going more and more into science, and I don't think I can be more science than this. This is, of course, this is the central dogma of molecular biology. So you have DNA, which is transcribed into RNA and which is transcribed into proteins. These proteins are either within the cell, or they are in the in the membrane, in the cell membrane, or they are outside of the cell membrane. And in fact, all the work, or most of the work we have done, or we did until the end of the 20th century in the field of molecular medicine, was targeting proteins. Why? Well, it makes sense because proteins are the ones who make all the hard work. So they are not only present in the cytoplasms, in all these chains, as kinases, as phosphatases. They are present in the membranes as receptors. They are present outside the membrane as hormones. They are also transcription factors. So in fact, the genetic code contains the instructions to make proteins, which are what are doing the job. So why not target them? We did that, and in fact, we were very successful. The problem is that there were some diseases or some targets which were not really amenable to treating them like this. And by the way, when I mean here target with capitals, I mean target in this scheme, not a biological target the way we normally apply to the term to. So if we look at this molecular dogma, then why not change this target with capitals, and instead of targeting proteins, target DNA? And this is, of course, what we are doing with gene therapy. If you have one missing protein because you have a mutation and that protein is not working, why not just making the body generate that protein? So yeah, this is one of the, let's say, probably the most clear-cut example of gene therapy. There are more. Then epigenetics. So if we think about the genetic code, 2% of our genetic code contains these instructions to generate proteins, up to 50-60%, according to how you define it, is devoted to controlling when to generate one protein. So it's devoted to sort of letting the environment influence the expression of the proteins, and also depending on the type of cells, of course, making the proteins that you need. So we discovered epigenetics. In epigenetics, we have the histones, the proteins, the DNA is around, it's wrapped around, but we also have the changes in methylation, which suppress or activate the expression of a gene. And this has generated lots of new targets with small letters, which we are now using to treat several diseases. That's DNA, but why not RNA? RNA is there in the middle. It's the disadvantage is that it's not as stable. The advantage is that it basically contains all the code that is going to be transcribed and then translated into proteins. So we can target that. Not only that, and you all know that we have BioNTech, we have Moderna. They have been hugely successful with the COVID vaccines. They are vaccines that are based on RNA. So basically, they are administering the RNA, and this RNA, which creates the spike proteins of the coronavirus, then the body is going to react and create an immune response to that spike protein, which is encoded by the RNA, and then it's going to create that immunity that is basically, in the case of all vaccines, it's basically taking us out of this pandemic. But not only that, so you have RNA therapy as well. So we are using antisense oligonucleotides. You are using siRNAs, where we are, or even microRNAs, where we are blocking, knocking down the expression of some proteins specifically. And this is another new field of therapy. And not more than that. So up to now, I've been talking about biomolecular entities, but why not use the whole cell as our tool, as a valid? And this is what we are doing, of course, with cell therapy, which is my next cartoon. With cell therapy, we are using the whole cell as the drug, really, as what we are getting into the body to cure, to heal that disease. And that can take, regenerative therapy, started by that, and now we have the examples like CAR-T therapy, where we are using the cells as drugs. But even more, so up to now, I've been moving in the different levels of organization of matter. I started with the molecules, I moved to the cells, and then, why not organisms? Why not use old enemies as allies? And this is something we have been doing for many, many centuries. This is a leech which was used for bleeding, and this is based on an old theory by Aristotle of the different fluids in the body. The theory was wrong, but Aristotle was right in using research and getting knowledge and building innovation on top of knowledge. So I'm not saying any bad thing about Aristotle, but anyway, this was applied, not very successfully in most cases. Then, of course, we took advantage of the products of some bacteria and archaea that had been creating them along evolution to fight other bacteria. This is the bacteria, and in this case, these were products by fungi that were created to fight bacteria. This, of course, penicillium, which created the first antibiotics. We used also viruses at that stage to create vaccines. So the first vaccine by Jenner, the smallpox vaccine, was an inactivated form was cow smallpox, or then inactivated forms of smallpox. And then now we are using microbiome. So all these 1 kilogram of bacteria that on average we have mostly on our stomach, and we are trying to use them to our benefit as well. And of course, I mentioned CAR-T before, and also genetic therapy. So for gene therapy, what we are basically doing is using viruses. We are using viruses because they have specialized in inserting genome into cells. And when we want to insert genome, what do? We co-opt viruses, and we use them for our own benefit. In the case of CAR-T, we use them to modify, to genetically engineer T cells, and those T cells then become the weapons that we are turning back into our bodies. But not only there, so if I think on what we could call the classical DD, drug discovery, which is a small molecule, which depends on this cycle of designing new small molecules, synthesizing them, testing them, then this was the old paradigm, and we have all these things in synthesis, automation, flow chemistry, DNA-encoded libraries, and then in the design, things like PROTACs or artificial intelligence machine learning. So what are PROTACs? Instead of blocking a drug with a small molecule, we have small molecules that basically label a protein by linking them to a ubiquitin ligase. They label it as to be destroyed. So this is an alternative way to block the effects of a protein in the body. And it has the advantage that the chemical structural requirements of the compound to become a PROTAC of a molecular glue are different to the ones of an inhibitor. So this opens really the chemical space. Artificial intelligence, machine learning, of course, applying all the advances on big data to predict and to design new compounds that will be active. DNA encoded libraries. So on the one hand, using CombiChem to generate really huge libraries, widening the chemical space. On the other hand, labeling them by a genetic label, which makes it easier then to identify which is the active compound. And yeah, finally, automation, which means that we are applying also technology and all the advances in technology to make more compounds. And in that sense, coupled with the advances in design, increasing the chances that we are going to find an active compound or a compound that will be active in humans. And things like flow chemistry, which was well known in the manufacturing, but not so well known in drug discovery, and that is now being used. This provides, on the one hand, a way to keep everything controlled, because everything is in tubes and it's only mixed the time that you want. And it only allows you to perform green chemistry, because everything is kept under closed conditions. And at the end of the day, what you isolate is the final, ideally, what you isolate is the final compound in the drop, and everything has been under tubes and not going to the environment. So, and that was, I believe, my last slide. So I think we have covered. We have gone a long way from the first guy that we saw that was using spells to cure diseases. We have built on science and on knowledge to create these new solutions. We've been very successful, but we need to be more successful in the future. And this is what we are trying with all these methodologies. And if you remember from that was actually my first slide, where I was from the corporate deck. I was talking about science and ingenuity. There was another component, which was heart. And this is what this quote from our founder, from Paul Janssen, was about. The patients are waiting. We cannot forget that what we are trying to do is to generate new solutions and basically to help patients overcome all these difficult diseases. And with this, I think I will finish. So thank you very much for your attention. And yeah, I'm open to questions.
Hi, Antonio. Thank you so much for your stimulating presentation. You have touched so many critical areas, which is solid need of the hour. And I'll ask the audience that if they have any question, they can ask you in the QA session or in the chat. Okay. Can you hear me?
At least I can hear you.
Okay.
I can hear you, and I enjoyed your presentation very much. It was, I felt like I had journeyed a lot longer of that drug discovery history than I should have. And I welcome the idea that things are moving a lot faster these days.
Yeah.
That's what we all are trying to do.
Okay, so thank you so much, Antonio. And if someone will be having, they will be asking any questions, they will be asking you in the QA session. You can check that, okay?
Okay.
Thank you so much. Have a great day. Stay safe. Bye. So, ladies and gentlemen, our next presenter is Vital Rottenbeck. She is with us. Just wait for a second.
Hello.
Hi.
Hi, hello everybody.
Hi, Vital. How are you?
Fine, thank you.
That's great. Okay, so ladies and gentlemen, Revital Rajenbeck, she is the CEO and chairwoman of 4P Pharma, and she's a serial entrepreneur passionate about science. After obtaining her PhD in biology and her MBA, she founded 5 biotech companies, including 4P Pharma in 2014 and 4 Living Biotech in 2020. Driven by a deep ambition to breakthrough discoveries from academic research to the patients, Revital ran 3 entrepreneurial adventures, of which she is at the origin. So, ladies and gentlemen, let's welcome Revital. So, Revital, thank you so much for being
Thank
here.
you.
And you can start your presentation now by sharing.
Yes.
Yeah, I can see your slides
Do
now.
you see my screen?
Yeah.
Great.
Okay, so good luck. Stage is all yours now.
Thank you very much. Thank you for the invitation first. What I'm going to present to you today is the how to accelerate development, pharmaceutical development by gathering together academics, labs, pharma company, and our expertise and experience in terms of developing fast pharmaceutical therapeutics. So first, I will present to you 4P Pharma and what we are doing. So what we are leveraging on marketed drugs, not because of the chemistry of the drug, but on the target that the drug that the drug action on. So our first thing is that we are a science-driven company. We are putting together mechanism of action of different molecules, and we link them to the physiopathology of different diseases, and particularly the diseases that are age-related diseases, such as osteoarthritis, neurodegenerative disease, macular degeneration, and also the severity of infectious diseases. So I will show you how we are doing to translate this science from the academic world to a product for the pharma and for patients. So we are sourcing all our therapeutics candidates in the academia. So we are working with different universities. We are working with hospitals and research centers where the science is done, where the discovery of the candidates and the mechanism of action of these candidates are discovered. So once we identify these therapeutic candidates, we are putting them in our 4P Pharma development engine. This is how we call it, is in our labs, and we are doing like a technical due diligence on these candidates, meaning that we are going to do preclinical development, in vitro and in vivo proof of concept. And once this proof of concept is done and the candidate is selected and it is ready to clinical development, human proof of concept, we are going to spin off the asset. Why are we doing that? We are doing that to have a single product vehicle in which we are doing those clinical trials, and this single product vehicle enable us to have pharma collaborations, and this is very important to bring this product to the market and bring this product to the patients, because pharma companies, we are using them as manufacturers and also as the entity that will commercialize our candidates and this targeted drug. So from the beginning, so we are located in Pasteur Institute at Lille for the lab, and in Paris we are at Institut de la Vision. And we have, from the beginning, we have screened for more than 300 candidates on the scientific rationale and the IP. From the 300 candidates, we have selected and evaluated 21 candidates with by doing preclinical studies and using our R&D capacities. And from these 21 candidates, we have selected for clinical development 3 candidates. And I will talk today on 4P004, which is in one vehicle for moving biotech, and also 4P021, which is the candidate for the fight on COVID-19 pandemic. So we have in our pipeline, so those 2 candidates that I will talk about, and we have also an early stage development pipeline. Again, what we are interested in is on the molecular target, the cellular target. So for the 4P021, the target is CXCR4, and I will show you how we get on this target. What is the scientific rationale? GLP-1 receptor is also another target for osteoarthritis. And again, I will show you those. I will make a focus on those 2 molecules. So first, for Moving Biotech, it's a spin-off company of 4P Pharma for osteoarthritis. And just to give you some science about what happened in the physiopathology of osteoarthritis, is that what we know is that there is an increasing inflammation with different markers. We know that there is also the matrix destruction of the cartilage with that can be shown by using different catabolic markers like metalloproteinase, etc. And we know also that there is a cartilage degeneration with a decrease in different markers, 2 markers called aggrecan and called 2A, that induce this cartilage degeneration. Osteoarthritis is concern like 300 million people in the world, and it is a very painful disease. And today there is no treatment at all. And this is what we are interested in: diseases that where there is no treatment at all. So the process of our development came from first a scientist. So this discovery came from the lab of Professor Francis Berenbaum, who is the head of rheumatology at Saint-Antoine Hospital, and he discovered a very important thing, and it is a new paradigm on osteoarthritis. Because before his discovery, it was osteoarthritis was an age-related disease, and that's all. And what he has shown is that osteoarthritis is linked to metabolic disorders. And so, as a GLP-1 analog have demonstrated anti-inflammatory properties in diabetic patients, he thought that those GLP-1 analog could constitute a new treatment for osteoarthritis. And we started our collaboration from there. And so what we have done is we have done different in vitro proof of concept using chondrocytes, which are the cells of the cartilage. We have shown that there is an anti-inflammatory effect of the GLP-1 analogs on this chondrocyte. We have shown in vivo that this anti-inflammatory properties lead to analgesic properties. And afterwards, we have also shown that there is a protection of the cartilage and also regeneration. So all this development has been done directly with the academic lab of Professor Francis Berenbaum, and we have today 3 patents on this indications, and we are now starting the clinical trial. So as I told you, in the models that we have used different models, not only the models for inflammation. We have also shown the same mechanism of action in models like surgical models, and we have shown that the 4P04, which is a GLP-1 analog, has anti-inflammatory properties, anti-catabolic properties, and also pro-regenerative properties. So in these 2 last years, we have done a lot of studies, and so it's just to show you that it is possible by using different collaborators and different partners to have to make the proof of concept and to go very fast into the clinical trials, and it can be done in 2 years and not in 5 or 7 years. So here we have in these last 2 years we have shown that we have done the proof of concept for the long-term pharmacology, structural efficacy. We have done a pharmacology dog in dogs clinical pilot study, modelization of PK/PD, CMC pre-formulation. In terms of regulatory development, we are now in CMC and GLP tox studies, and now we are preparing the pre-CTA for European Regulatory Agency to go to the clinics. So this is our lead asset before the COVID-19 pandemic. And we leverage on this development and on how we and our knowledge on how to go fast into the clinics, and we started the development of our CXCR4 antagonist for the treatment of acute and chronic complications related to viral infections, and particularly on COVID-19. And we did it very fast since the discovery came from start in May 2022. So what we wanted to address with this 4P021 is the acute and the persistent lung complication related to COVID-19 and other viral infections. So at the moment where we started this development, we knew that in hospitalized COVID-19 patient, there were prolonged pulmonary complications that lead in 60% of those patients to lung fibrosis. And so what we wanted to target is to ameliorate the respiratory function and the hospitalization under respiratory assistance, and also the long-term pulmonary lesion. And to do so, we wanted to address the immune system and not the virus by itself. So I will put you in the context of April 2020, because this is how we get to this scientific rationale. So at this moment, the pathogenesis of COVID-19 was described as inflammatory damage of bronchial wall. There were some papers, there were a lot of papers and a lot of literature on COVID-19, but there are some papers that have shown that the pathogenesis was similar to the high altitude pulmonary edema. And there was also a proliferation of fibrous tissue and fibrosis. So from those descriptions, we have thought about the mechanism of action that can be targeted for this COVID-19 pandemic. So there was, on the high altitude pulmonary edema, we thought about the neutrophils that can be a good target for the migration of the neutrophil to the lungs, could be a good target for COVID-19. And another paper from 2016 that came from CHU of Bordeaux, and we knew this Professor Patrick Berger, who is a pneumologist, and he has shown before that the blood fibrocytes, which are immunocells that are between monocytes and fibroblasts, were recruited during acute exacerbation of COPD. So by putting together those paper and other, of course, literature data, we came to the hypothesis that CXR4 can be a good target for COVID-19 severe patients. So this is how we filed our first patent on this CXR4 as a target. And then we have started the in vitro and in vivo proof of concept. And we have shown that by using a CXR4 antagonist, we inhibit the neutrophil migration, we inhibit the netosis, but I will enter a little bit more in the mechanism of action. And we have also shown that there is also a decrease in pulmonary fibrosis using this CXR4 antagonist. And during this time, our problem was the manufacturing of the molecule, and to have a good CXR4 antagonist that we can use in the clinics to accelerate the development and to bring this asset to the patients. So we started to discuss with a big pharma partner, and that was that helped us to manufacture the product and also the placebo. And now we are launching the phase 2B clinical trial. So to enter a little bit more in the mechanism of action, which is very important in our development, we after our discovery, there were several papers on CXR4 involvement in COVID-19 severe patients. And particularly a team from Gustave Roussy Institute in France has published that by comparing mild patients and severe patients, they have shown that there is an emergency myelopathy in severe patients, and that there is an accumulation of immature neutrophil which are CXR4 positive, and and with the other papers that are in the literature, the hypothesis was that the migration and the accumulation of this neutrophil induce into the pulmonary interstitial tissue, induce the acute phase of COVID-19. And moreover, this CXR4 neutrophil also are forming what we call the neutrophil extracellular traps, netosis, and this netosis induce the recruitment of other cells like the platelets, and this lead to microthrombosis, and also to the recruitment of fibrocytes, which lead to the fibrosis in the lungs of severe patients. And so we have done all the proof of concept, as I told you, in vitro and in vivo. And what we have shown is that when we're using a CXCR4 antagonist, we decreases the neutrophil migration induced by COVID-19 patient sera. We also decrease the netosis of these neutrophils, and we improve also the acute and long-term lung pathophysiology in a mouse model, and particularly the lung fibrosis. So from the target product profile of this CXCR4 antagonist is to target first immunosuppression, as I told you. And there is a very recent paper, again, that is from 2021, showing that there is an increased proportion of inflammatory CXCR4 T cells in the lungs of individuals with severe COVID-19. And this increase in CXCR4 positive T cells is correlated with the morbidity in severe patients, COVID-19 severe patients. So our CXCR4 antagonist will target the immunosuppression and also prevent long-term lesions. So this is our hypothesis, and this is what we're preparing today to show this in a human proof of concept. So as I told you at the beginning, to develop it, to make this possible, we have to gather together different entities, not only a team that is the internal team of 4Living Biotech, with people that are medical doctors and also regulatory people and also market access experts. And so to do this in 18 months, we have worked with the University of Hospital of Bordeaux, the Pasteur Institute of Lille. We worked also with the regulatory entity, with our R&D capabilities in-house at 4P Pharma. And of course, a clinical CRO to make this clinical trial possible, and a big pharma that help us and support us to have the manufacturing of the product and the placebo. So to summarize what we have done in a very fast process, is that we started from a scientific rationale that was at the beginning of the COVID-19 pandemic, and where there were a lot of papers and literature data, not always peer-reviewed, and even with that, we by leveraging on our partnership with academics, we were able to support our hypothesis, which was the importance of CXCR4 in the correlation with the severity of COVID-19. This was our hypothesis. So we have done preclinical POC, 2 patent filing during this time. We have done this clinical research agreement with the big pharma, and that help us also for the design of the clinical trial, of course. And today we have the IMPD that is ready, and the clinical trial is scheduled in 6 countries. And first patient in is planned for the end of this year or at the beginning of next year. So to summarize what we are doing in 4P Pharma and how we accelerate this development is first that we building scientific approaches by linking innovative cellular target to disease biology. So I gave you the example of GLP-1 analogs. I gave you the example of CXCR4 antagonist, and we have other examples that are in our pipeline. And it is very important when we work with key opinion leader, that academics, is to do this technical due diligence, meaning the preclinical studies and in vitro in vivo proof of concept in a laboratory with very standardized processes. The second thing that we are doing is that we leverage on fast track, on regulatory fast track to target those large unmet medical needs. So, for example, going to developing an asset, a candidate, a therapeutic candidate for influenza or other viral infections. To do that, we let's say we leverage on the COVID-19 regulatory fast track to go very fast to the patients. For osteoarthritis, what we know from 2018 is that today the FDA considered osteoarthritis as a serious disease, and there is a fast track and potential conditional market approval for drugs that show symptomatic effect, a very important symptomatic effect, and also that have shown some structural effects. So there is a possibility for us to go very fast to the patients by going to these large unmet medical needs. And finally, how we reach the patients and how we bring our development, our candidates to the patients. And to do so, as we are a small biotech company, we are not going to commercialize any of our assets. And so what we're looking for is a big pharma collaboration. So first, of course, for the manufacturing of drugs, because the proof of concept, we are doing them with drugs that are already past the phase 1 or phase 2 in other indications. And so we are doing this risk mitigated path to market by our collaboration with the big pharma. And so we have done that for Living Biotech. So today we have this collaboration clinical agreement. And for Moving Biotech, we are now starting the discussion before we are starting to the phase 2 clinical trial. So I'm done here. So thank you very much for your attention, and I will be happy to answer to your questions.
Hi, Revital. Thank you so much for your wonderful presentation. And I'll ask the audience that if they have any question, they can ask you in the QA session or in the chat session. Thank you so much for being a part of our conference and adding value there. We are looking forward to work again with you.
Thank you very much.
Have a great day. Stay safe. Okay, so ladies and gentlemen, our next presenter is Fred Jacob, who is working as a CEO at TYT Oncology. And he is Fred Jacobs. Hi, Fred, can you hear me?
Yes, I can.
Can you please start your, yeah. Hi, how are you?
I'm fine. How are you doing?
I'm good, thank you.
Thank you for inviting me and for Platinum Global and the opportunity to speak today.
Thank you so much for being a part of this conference, and you can start your presentation by sharing your screen, and I'll ask the audience that if they have any questions, they can ask you in the QA session or in the chat session during your presentation. Okay.
Are you able to see my screen?
Not yet.
Okay, well, geez, I have too many windows open. Okay.
Yeah, it's working now.
Okay, terrific. Well,
Okay.
thank you everyone for your attention, and it's kind of appropriate that I'm the last speaker before lunch because gastrin, which is the main thing that I'm going to talk about today, is something that has a lot to do with our thoughts about food, when we smell food, when we look at food. There's all kinds of different ways that gastrin is triggered in our bodies. And for many years, we knew that gastrin was a culprit in many different cancers. It has driven cancers from the esophagus straight on through the gastrointestinal tract to the colon, and includes the lungs in between. And we just never really had a great way to treat it. And I entitled this presentation let's see if I can get the slides to advance. I entitled it The World's Most Dangerous Hormone, and many people would not guess that gastrin is responsible for more disease and death than testosterone or estrogen, who are usually chosen as the most dangerous hormone. Gastrin is the original growth hormone. When all of us were in our mummy's womb, gastrin crossed the placenta from mom and was used to grow every major organ in our bodies. It is in the third trimester that is switched off. And when you're an adult, if you have gastrin receptors and gastrin in your pancreas and other organs, it probably means that you have cancer, because although gastrin is instrumental in helping us to grow and become young people, like many hormones, including testosterone and estrogen, later on in life, these same hormones that were so instrumental in early life become dangerous. And one reason that gastrin in particular becomes dangerous is that everybody's gastrin levels rise as they age. And there's a couple of reasons for that. One of them is H. pylori infections. H. pylori infections are strongly linked to stomach cancer in particular. And H. pylori is known to facilitate high serum gastrin levels, particularly of one of the members of the gastrin family of hormones, G17, named because it's 17 amino acids long. So if you are one of the people in the world who has an H. pylori infection, then the top quartile of that group are the ones who have the highest level of hypergastrinemia. They're the ones who are most at risk for developing cancers and other deadly diseases. Similarly, if you're taking proton pump inhibitors, then you have hypergastrinemia. Proton pump inhibitors work by inhibiting the acid, and in doing so, they create a hypergastrinemia in response. And those patients that have the highest quartile of serum gastrin levels will be the ones who are at most risk of developing cancer. And what our lead product is really a patient-friendly, low tox, low cost therapeutic vaccine against that. We go after not the entire gastrin family of hormones. We go after just two hormones, G17 and its evil cousin G17 glycine extended. These two hormones are most of our gastrin at any given point in time. And this is such a low tox drug. It's administered by subcutaneous, is designed to be administered by subcutaneous injection, that we think that it can be used by home health visitors to treat patients, to help patients avoid hospitals, which is particularly important in this day and age. So there's a lot of publications out there, but this 2018 study in Korea, which has the highest risk of pancreatic cancer for adults anywhere in the world, and not coincidentally also the highest smoking rates for adults anywhere in the world, showed that there was a relationship between proton pump inhibitor therapy and pancreatic cancer. And so we think that this is something that is probably going to happen more and more as we age, that things that were so essential to us when we were young become maybe troublesome to us as we get older, and we need to control these levels. So in terms of drug discovery, we think that what we're doing, which is we call active checkpoint control immunotherapy, is better than Nobel Prize winning monoclonal antibodies and better than second generation checkpoint inhibitors. And the reason for that is that we harmonize with the immune system, and we provide the patient with a very high quality of life under treatment. And we're hoping that we're going to provide the patient with very low or no risk of side effects. In our case, if we're able to go after a transmembrane target, we have the chance to simultaneously induce CDC, ADCC, and cytotoxic T cell killing, which gives us a chance to actually be curative in some of these cancer cases. So how do we do this? The problem with an earlier version that we used was that we were using diphtheria toxoid to try to deliver as a protein carrier our antigen into the body. And it was not strong enough. It was not immunogenic enough in order to overcome self molecules. The immune system sees gastrin every day. It's ubiquitous in the system. The gastrin receptor is perhaps the most prevalent receptor in the body, and goes right up through the CNS. So it's how do you then take something that the immune system is completely used to, sees all the time, and specifically target that one or two members of the gastrin family of hormones and leave everything else alone? Well, it comes really from the serendipity of our chief science officer and his discoveries. And this is really his slide, and I'm not going to try to explain it for him, but really the idea is that we're able to activate and recruit B and T cells to produce huge amounts of antibodies. And in my personal case and in my personal journey, this allowed us to target gastrin, which we knew needed to be targeted, but we just didn't have a strong enough protein carrier to make it work. And that all changed when we started using this active checkpoint control immunotherapy. So what we're doing is trying to overcome anergy by using TLR9 in conjunction with a very specific antigen, and using this platform is like a plug and play. So if in your drug discovery efforts you have found a target, but you don't have the right SpaceX rocket for it, then you should get in touch, because we might be able to help you. This targeting system will go after just about any water-soluble protein. And as I say, we're able to trick the immune system first by activating T cells. We use a CD32 connector to allow the TLR9 to be internalized into the plasma dendritic cells. Coincidentally, CD32A is where we dock on the plasma dendritic cells on the activated B cells. It's CD32B. And yet we were able to use CD32C to make both ends work. It was just kind of a serendipity thing. And once we get the warhead into the cell, we have these activated B and T cells, and the IgG response is amazing. We would love if some of you out there have experience with IgG response, if you would share with us some of the responses that you've had in your trials, because we would like to have some measure of comparison, and I'll get to those levels in just a minute. With TIG-300, I mentioned that we can unleash the full immune system complement by going after transmembrane cells. And here we're going after the gastrin receptor. Most people are familiar with CCKA and CCKB, but there's also a CCKC for cancer. It's a little bit bigger gastrin receptor. It has 60 some odd additional amino acids, and it's capable of producing its own gastrin that it uses to fuel its activity. This is how the autocrine mechanism works in pancreatic cancer. So I mentioned before, if you have gastrin circulating in your pancreas, you probably have pancreatic cancer. And there is a test out today called GastroPanel that measures G17 levels, and it measures H. pylori antibodies. So we had nothing to do with the inventors of this medical device, but we applaud their efforts because they've basically identified the patients for TIG-100 for us. All we have to do is ask them if they're taking PPIs too, and we've got our patient. So here you can see how we're able to activate the killer cells and go after the tumor in with a transmembrane target. This is able to bring all of the innate and adaptive defenses of the immune system together simultaneously. And we hope that this means that there is no chance for the tumor to escape. We're hopeful of getting to the clinic with TIG-100. We are working with a company named Apolomix that has recently taken the rights for some of the world, and they're negotiating for some additional world rights. And they've been a very good development partner, and we think that they will work with us to a very high standard. We're going to target both stomach and pancreatic cancer initially because they are both driven by G17. If you in recent years, there are publications with the name gastrin and pancreatic cancer and gastrin and gastric cancer. And in the case of gastrin and gastric cancer, there were two different authors on two different sides of the Atlantic Ocean who came up with the same catchy title. And approached the subject differently, but came to the same conclusion that gastrin drives stomach cancer. We have a U.S. orphan drug designation for the U.S., and we're hoping that that will help us get to patients sooner. And I'd just like to show a little bit of results that we have comparing the old formulation, which was named G17DT, with TIG-100, our lead product. So on the far right, you see the lowest columns, and they represent phase 3 clinical trials that were conducted with the previous formulation. PC4 was a combination trial study involving G17DT with gemcitabine against gemcitabine alone. And PC6 was a monotherapy trial, which in all of the trials, and the predecessor company conducted 22 trials in gastric, colon, and pancreatic cancer. In all of those trials, any patient who had any polyclonal antibodies developed against G17 had a positive correlation with survival. And it was antibody dependent in how well their survival was. The problem with the old formulation is that most patients didn't make many, and you can see that the average was below 16 units. If you had 16 units, you had better survival than chemo or placebo. And you can see that the average fell short because of so many non-responders. The USA study in the middle column that was first done with the first formulation, we were just so ecstatic because here we had 100% responders, 100% high responders, and the titers of antibodies were reached in just a few weeks. 0 side effects. This was very well tolerated by non-human primates. The previous formulation was well tolerated by humans. We thought that this is going to be gold. We did some additional studies, and we repeated the first study, and then we did 1/5 of the dose and 5 times the dose to get an idea of dose ranging. And on the far left, you see with just a 6 microgram dose, we were able to generate 100 units compared to less than 16 with the old formulation. And we repeated the US study and got similar results. And then we did this 154 microgram dose. And that's where we got this IgG response that was really off the charts for us, which I'll come to in just a minute. But the group that I was actually most proud of was this vertically striped group, because here, these non-human primates received gemcitabine before receiving TIG-100, and they still produced exceptionally high titers of antibodies with the original dose, although there was a lag of a couple of weeks in reaching those kinds of levels, which you might expect if someone's taking chemo, which impacts the immune system. So here's where I'm talking about the very high IgG, anti-G17 titers. We're getting 150 micrograms of IgG after only 2 weeks. And we think that this is exceptional. And again, we have 0 side effects. We're working diligently with Apollomex to develop GMP material suitable for humans, and we have key opinion leaders who are standing by to try to deliver for us. So I'm just about out of time here, and I'd like to leave a few minutes for any questions. And I also wanted to comment that to Antonio, if you're still listening, that J&J was an early investor in gastrin neutralization. They were a 10% minority shareholder of the predecessor company, and when they received their shares, they sold the stock at a good profit. We were publicly traded, and they were allowed to, and we couldn't get them to invest again. But if you're interested in geographic rights for any of the countries, do get in touch. And I was very impressed also, I wanted to say, with 4P Pharma and the road that they took and what they accomplished. And something similar to that is what we're probably going to want to do with this CCKC, this intron C variant of the gastrin receptor. So maybe there's a chance for collaboration in the future. Anyway, any questions? I'm happy to take them. If not, you can go early to lunch.
Hi, Fred. Thank you so much for your wonderful presentation. And I'll ask the audience that if they have any questions, they can ask you in the QA session or in the chat session. Okay, can you hear me? Hi, Fred. Hello. Can you hear me? Hello. Can you hear me?
I can hear you.
Okay. Okay, so thank you so much for your wonderful session. And I'll ask the audience that if they have any questions, they can ask you in the QA session or in the chat session.
Okay.
Okay, thank you so much for being here. Have a
Thank you.
great day. Stay safe. Bye. Okay, so ladies and gentlemen, now we will be having a short break for 15 minutes. And after that, we will be back with Moza Yodium's presentation. And you guys can have a break. And let's get back after 15 minutes. So you all can grab your coffees and lunch and anything to eat. So we will be back. So now our next presenter is Moza Yodium. He is with us. Let's welcome him. Hi, Moza, can you hear me? Hello?
Can you hear me?
Yeah, I can hear you. Can you hear me?
Yes, thank you.
Okay, kindly start your video. Hi, how are you?
Hi. Very well, thank you.
That's great. Okay, so Moza, thank you so much for being here. And ladies and gentlemen, this is Moza Yodium. He is President, CSO, and Professor at Emeritus from Yodium Pharmaceuticals. And he's working as a CSO and he's working here since 2012. So Moza, thank you so much for being here. And you can start your presentation now by sharing your screen.
Okay.
Yeah, great.
First of all, I'd like to thank you for inviting me. And good afternoon, everybody. We are a very small pharmaceutical company, but we think we are highly innovative. And I'll be talking about multi-target drugs for the neurodegenerative diseases. And we know that at least in Alzheimer's disease, Parkinson, multiple sclerosis, ALS, and Huntington's disease, there have not been any drugs that have disease modifying. They're all symptomatic treatment. And as a consequence, we've been trying to develop drugs that will have disease modifying effect in these diseases. Specifically, in Parkinson's disease and Alzheimer's disease, can be developed neuroprotective drugs that can protect the neurons, and neurorestorative, which is even more difficult. We heard earlier that some of these diseases start way before we actually see the symptom. At this, in Alzheimer's disease, we think the disease may start 15 or 20 years before. And in Parkinson's disease, about maybe 5 to 10 years. What is interesting is that we know that our neurons in the brain are dying at the rate of 3% every decade. But in Parkinson's disease, when we look at Parkinsonian brains, which we have examined through the years, we know they have lost about 75% of their neurons, and that's why they develop the disease. Therefore, if you live long enough, they get Parkinson's disease or Alzheimer's. So age is a major factor in these diseases. And what we are trying to do is to really develop drugs that can protect the progression of the disease or develop drugs that have restored the neurons. Both Parkinson's disease and Alzheimer's disease are complex multifactorial disorder, and it's been naive to think that we can develop one drug that can treat the disease of Parkinson and Alzheimer. Here it just shows you the cascade of events that goes on in Alzheimer's brain. Very similar thing happens in Parkinson's disease. We have, but of course, the amyloid, we got the inflammatory, we got mitochondria, we got oxidative stress, we got accumulation of iron, which I talk a lot about. And many factors are involved. There's this cascade of neurotoxic event that goes on. And through the years, we've been studying these diseases, and the pathology from brains of various neurodegenerative diseases like Alzheimer's, Parkinson, Huntington, amyotrophic sclerosis has shown that there's a cascade of events and there's a homology among these diseases. That means we see the accumulation of iron at the site where the neuron dies. There's nitrogen species, inflammation, glutamate toxicity, mitochondrial complex, ubiquitin protein system dysfunction, abnormal protein function, all going towards oxidative stress. Was a component that Peter Reeder and myself discovered back in 1989 and has remained really the center of these diseases, oxidative stress and metabolic impairment that eventually lead to mitochondrial collapse that result in death. So we know that mitochondria is important for cell survival and cell death. And one of the things we've seen in animal studies that confirm what we have seen in the pathology of the human brain is that where the neurons die, there's accumulation of iron. And iron plays an important role in formation of free radicals that can give oxidative stress. This is the model of Parkinson's disease, 6-dopamine that lesions the dopamine neurons into the substantial neural pars compacta, iron accumulates. Here's lactocystin, another model of Parkinson's disease that iron accumulates. This is the model of the genetic model of ALS. Again, we see in the interneurons in the spinal cord, there's an accumulation of iron. And this is kainate model of Alzheimer's disease. You see the iron accumulates in the dentate gyrus and in the hippocampus. So iron has become really an important component to develop drugs. The other thing that's absolutely of paramount importance in that amyloid precursor protein and some psychonuclein that have been implicated in the pathology and in Parkinson's, in Alzheimer's disease and in Parkinson's disease are iron-regulated protein, are identical to ferritin. They have this 3', 5' in their mRNA, and we know that iron regulates APP as well as alpha-synuclein and of course ferritin. So iron became an important component for us. And the concept of oxidative stress in both diseases, Parkinson, came from the fact that we know that the monoamine oxidase, which is a mitochondrial enzyme that reacts with amine to produce hydrogen peroxide, may play an important role in the pathology of disease because this enzyme increases in gliosis in Parkinson's disease and Alzheimer, and we know that iron accumulates, so iron interacts with the hydrogen peroxide in the Fenton reaction to give you the highly toxic radical, oxygen radical that results in the cascade of in oxidative stress that eventually lead in cell death. I put apoptosis here because we really don't know how the neurons in Parkinson's disease, namely the dopamine neurons in the Niagara die or the cholinergic neurons in the hippocampus. So we started, I've already developed three drugs. One is called the Azilect for Parkinson's disease. It's a monoamine B inhibitor. This molecule has a very interesting thing. It has both monoamine oxidase activity as well as it has neuroprotective activity in vivo and in vitro. It's already been approved by the FDA for some years. And we discovered it's this proprietary moiety that has both the MAO inhibitory and the neuroprotective activity. We had developed a new iron chelator called VK28, and we decided what we will do, can we put these moieties, the propargylamine, the monoamine oxidase inhibitory activity, and the cholinesterase inhibitory moiety that, you know, the cholinesterase inhibitors are used in the treatment of Alzheimer's disease like ribostigmine and donzapil, the carbamide moiety into iron chelator and develop a multi-target drug. And indeed, we were very successful. We have patents on all of these drugs. on all of this compound, and we were able to put this propargyl first in modified VK28, the compound M30. We went and put it into another with the propargyl moiety, HLA20, and then we put the carbamide moiety into M30, and these drugs have really unique properties. For example, M30 is an ion chelator, radical scavenger, is a brain monoamine oxidase A and B inhibitor. It has antidepressant activity because of its monoamine oxidase inhibitor, and it has also cognitive enhancing. Even more so is this compound, because this compound has all the components of M30 plus the carbamate cholinesterase inhibitory moiety. So these are the concepts, and we started this back in 1994, developing multi-target, and many people are now copying this component of putting different moieties in molecules, which can be released from the main molecule to act, say in this one, to inhibit monoamine oxidase, and this carbamate gets released to inhibit cholinesterase, and then we are left with the ion chelator, which acts as an ion chelator and radical scavenger. So these are some of the compounds I developed. Rasagiline, I discovered selegiline with another monoamine oxidase B inhibitor. We went, and what we did, the first multi-target drug that we developed was to take rasagiline and take the carbamide moiety of rivastigmine Exelon and put it into rasagiline. So we developed a drug called ladostigil, which was a monoamine oxidase inhibitor, cholinesterase inhibitor, and it had neuroprotective and neurorescue activity. And we did a clinical trial with this drug in Alzheimer's patients. Unfortunately, the drug failed because we used a low dose. Somebody made a mistake about the dosage that we used, and we hope to go back and do a high dosage as a consequence. But I want to concentrate on my ion chelators, which are really, I think, are a very interesting component to it, because not only these drugs have neuroprotective activity, but in animal models of Parkinson's disease, they restore the neurons. This is really crucial. We know of no drug who's been developed by any drug company that can restore the neurons, and I'll show you later on about this. We have many other components. Now, this is our recent compound that we have now developed, and we have patent from Europe, USA, China, and Israel. We took rivastigmine, a very well-known anti-Alzheimer drug, which was developed by Novartis, and we put a propargyl moiety into it. None of the cholinesterase inhibitors presently in the clinic have neuroprotective activity or anti-inflammatory. What we discovered that when we put the propargyl moiety into M31, this became a cholinesterase inhibitor, of course, both inhibitors, became a selective MAO inhibitor, and it had antioxidant activity and regulation of neurotrophic factor, BDNF, GDNF. We are very excited about this. Really, this drug, we want to really develop this, go into the clinic with this particular drug. So this was one of our latest compound that we developed. As I showed you, these drugs have both monoamine oxidase inhibitor as well as cholinesterase inhibitor. It's really, and they have, as I said, monoamine oxidase inhibitory activity in the brain in different areas, and very little effect in the intestine and the liver. So we don't see the toxicity that we see with other drugs that increase brain levels of the neurotransmitter dopamine, serotonin, and noradrenaline. We know they play an important role in depression. All the drugs that we have today for treating depression act on these neurotransmitters. And they have antidepressant activity in the forced swim test, a classical test that is used for treatment. And we also showed that these drugs have cognitive enhancing activity. In the scopolamine, Morris Watermace, where you give scopolamine to rats, and they have to find a platform in the water tank. They are not able to, but when we treat them with M30 or M31 at 1 milligram, 5 milligram, after 4 days, they are able to find a platform very similar to the control. So we see, beginning to see that, yes, this multi-target drug is an approach, because most of the people who were trying to develop multi-target drug, where they were taking 2 drugs and putting them together. So they break up to the original drug, and they will have metabolites and then maybe interaction. What with this method is of multi-target drug, where we put the moieties get released, and the parent compound is also available to act. And we showed that this drug, at least the multi-target that we have, really do have memory retention activity in both in the transgenic model, and recently collaborating with Claudio Coelho in Montreal, who has the first transgenic rat model of Alzheimer's disease. They also show cognitive enhancing activity. And they regulate the amyloid precursor protein in the frontal cortex, hippocampus, and parietal cortex. So I won't spend too much time on that, but I want to go to something really the most important aspect of our work. That means neural restoration, neural rescue. One of the things that we discovered with our multi-target on M30 and the other ones is that when we cultured the neuroblastoma cell in absence of serum, and we put our compounds in there, they differentiated the cells, and GAP-TH, which is a neuronal thing, showed significant increase, showing that the drugs had neurorescue, neurotrophic-like activity. This is really crucial. This was a really amazing finding. We think this, as I said, we have never seen any other drug doing this particular type of thing. And we used embryonic stem cell in culture. You know, Israel is one of the center of embryonic stem cell, with the iron chelator, and it differentiated the embryonic cell into neurons. They contain GABA, glutamate, and dopamine when we culture these cells. One of the models of Parkinson's disease is the famous MPTP. When you give MPTP, it causes the degeneration of the dopamine neurons, but when you give M30, the iron chelator, it completely protects. And you know, in the MPT model, also iron accumulates specifically in the substantia nigra pars compacta, where the dopamine neurons die, and that give rise to Parkinson's disease. Now, one of the things that we did was to use the lactocysteine system. You know, the ubiquitin system is, we know that the misfolding of protein in Parkinson as with Alzheimer. And lactocysteine is an inhibitor of the ubiquitin system, UPS system. We did a study by giving first lactocysteine to the animal, and then waiting 24 hours, and we see that the neurons die. When we treated our animals, mice animals, with iron chelated, sorry, VK28, with VK28, I hope I can go back previous, VK28 and M30, it completely protected against lactocysteine. This is neuroprotection. We give the drug, then we give lactocysteine, it completely protects it. But you know, when we treated with lactocysteine, 24 days later, dopamine neuron in the compactor completely are degenerated. But the most amazing result that we found was that give the lactocysteine, cause degeneration, and then treat our animals with VK28 for 2 weeks or M30, and the neurons came back. Neural restoration. We have no other drug that we know has done this. We have a whole series of compounds that do this particular thing, and we have looked at this in several different models. The neural restoration we've seen in the 6-hydroxydopamine, in the MPTP model. So we become rather excited that this could be really a new approach for treatment of Parkinson's. This is the MPTP model. You can see the neurons are restored after the lesion with the neurotoxin MPTP. And we saw that there is actually neurogenesis in the hippocampus with our compound, suggesting that there is something about this, and we wanted to know what the molecular mechanism of this drug was. One of the things that we discovered on analysis of brain analysis, we were the first group in the world to use genomic transcriptomics to look at what happens in the degeneration of the dopamine neurons in the compactor. We used the gene analysis of 27 on gene chip. We discovered a gene called EGNN1 that is proline 4-hydroxylase. Proline 4-hydroxylase is an iron-dependent enzyme. Iron is its cofactor and is the key iron oxygen sensor in the brain. And it regulates the degradation of hypoxia-inducing factor. When the activity of proline hydroxylase goes up, there is degradation of hypoxia induced by ubiquitination. And it results in the decrease in angiogenesis, cell proliferation, glucose metabolism, and downregulation of the various neurotrophic factors that have been implicated in both diseases, in Parkinson and other, VEGF, BDNF, and GDNF. But a paper appeared that if you inhibit proline hydroxylase, you can reverse the concept that the neurotrophic factor would go down. And of course, it being an iron chelator, we use our iron chelator, and we showed that yes, the iron chelator inhibit proline hydroxylase. And what it does, it causes hypoxia-inducing factor to go up, and so did the neurotrophic factors. I'll show you the data later. So this is a drug that does not only have neuroprotective activity, but it seems to have also neuroreceptive action, possibly to the fact that it really activates hypoxia-inducing factor. And we saw this, by the way, this was the data from Parkinson in brains, where proline hydroxylase activity is increased. So we know that neurotrophic factors go down. Here it shows you that indeed our compound do induce hypoxia-inducing factor, both with the M30 and HL20. Now, what is so important about HIF? HIF plays an important role also in cancer. In cancer, there is cell proliferation, but hypoxia-induced factor activity is complex because it acts on the cell cycle. And our compounds cause cell survival and cell differentiation because we show that our compound causes cell cycle at G0, G1. That means the cell goes into cell differentiation and not cell proliferation. And I think that's a very important aspect of the gene. And as I should mention to you, that the hypoxia-induced factor is significantly increased by our compound iron chelators in this system. And you can see, indeed, BDNF, GDNF are significantly increased in brain areas in which there is neurodegeneration, suggesting that this compound really, if we get into the clinic, there's a good chance that it may really have some neurorestorative activity, at least in Parkinson's disease, etc. And this really shows that that hypoxia-inducing factor really does, through our compound M30, acts at G0, G1, and the cell goes in cell differentiation, and cycling D1 is significantly decreased. Now, in the final part of our talk, we did an analysis of Parkinsonian brain with a whole group of colleagues from the United States and our own group, where we examined 400 brain samples from Parkinsonian brain, specific the Niagara gene analysis. And what we found that the gene analysis suggested that there is a change in mitochondrial function, and that PGC alpha, which is really regulates mitochondrial biogenesis, could be approach to treatment of Parkinson's. We know that complex 1 and complex 2, 3 are defective in Parkinson's disease, and complex 1, I think, also in Alzheimer's disease. So having a drug that can actually activate PGC alpha could be an important aspect. So we've done a lot of work with our drug, and indeed we have discovered that both of our iron chelators really, which activate hypoxia-inducing factor, do activate PGC alpha, and that they do eventually cause biogenesis of mitochondria. I don't have time to go into the data that we have published with these results as a consequence. I want to go back to our first drug. One of the molecular mechanism in which our drug has neuroprotective activity relies on the propargylamine moiety. Everything that rasagilin, azilect, and M30 and ladostigil do in their neuroprotective activity relies on the propargylamine, and propargylamine does everything that these compounds do. And we've done the molecular mechanism that this propargylamine neuroprotectivity depends on activation of PKC alpha and epsilon and downregulating delta and gamma, and causing increase in BCLX and downregulating BAD and MAX, and the downregulating cleaved caspase 3. So that's the neuroprotective activity. of the compound. But the neurorestorative activity of the compound depends on, as I mentioned, to both APP and alpha-nuclein have this 3' 5' in their mRNA that can cause translation of APP and alpha-synuclein. It affects the proline 4-hydroxylase that resides in hypoxia-inducing factor to be increased that results in BDNF and GDNF and VEGF and erythropoietin. And of course, it also won't go into the regulation of alpha secreted that results in neuroprotection and neurorescue and neuroplasticity. We think this is really an exciting pathway. I'm not a chemist, I'm a neuroscientist, but I have a knack of developing drugs. All of these ideas came from learning my children doing Lego. I said, why can't we develop multi-target drugs by putting moieties onto compounds that we do? And many people now copying this concept of multi-target drug using moieties rather than combining 2 drugs together, etc., or giving 2 drugs. So we think this is a unique and novel approach for the drug design. And of course, we're not able to take this compound to clinic because we're a small drug company and we would like to collaborate with the larger pharmaceutical company that may be interested in some of the compounds that we have developed here in our compound. So in summary, an M30 has this, it has antidepressant activity because it inhibits monoamine oxidase A and B, and MAO inhibitors are antidepressant. They were the first antidepressant to be discovered back in 1960. They have anti-Parkinson drug because they increase brain level of dopamine. That's the act of Azilect or Rasagiline. Mentioned you that they are ion chelators or radical scavengers. They themselves increase GDNF, BDNF, and I'm sorry again, GDNF and BDNF for the neurorescue activity. Ion chelation results in inhibition of MAO and downregulation of H2O2 that prevents the formation of the highly toxic hydroxy radical. I didn't go into that they affect glutaminergic release. And I mentioned to you that they activate hypoxia-inducing factor that results in the BDNF. So we think this is a novel approach. I'm sure other people can think of developing other compounds with you putting specific moieties for other diseases rather than giving a combination of drugs which can interact, especially in Parkinson and Alzheimer. We know that both Parkinson and Alzheimer patients have responded quite badly to psychotropic drugs. So one drug doing the various things would be an important approach for the treatment of that. So finally, I thank you for listening to me.
Hello, Musa. Thank you so much for your wonderful session. Can you hear me?
Yes, of course.
Okay, great. Okay, so thank you so much, Musa. It was very wonderful session and it was really very interesting. I hope I would have learned so many new points from your presentation. And we are looking forward to work again with you. Thank you so much for being here.
Thank you.
Stay safe. Okay, so ladies and gentlemen, now our next presenter is Kjell Stenberg, who is working as a CEO from Actena. And I'll just ask him to join us for this session. He'll be with us within 2 moments, so stay connected. Thank you. Okay, so can you please start your video?
Oh yeah, I will start my presentation.
Okay,
That's clicking
sure.
on my video or
Okay.
sharing my screen.
Okay. So Kjell, thank you so much for joining us. And ladies and gentlemen, Kjell is working as a CEO from Actena, and he's working in drug discovery industry since 1975. So he's having 46 years experience in this field. So it would be very interesting to listen to his presentation. So Kjell, you can share your screen.
I will.
Yeah,
And I will take away that one.
Okay.
And I will be.
Yeah,
and
that's
how do I
great.
do?
Yeah, thank you so much for being here, and you can start your presentation. Thank you so much. Stage is all
Wonderful.
yours.
Thank you. Thank you very much for your kind introduction. I'm going to talk under the table Accelerating Drug Discovery by AI, meaning computerized support and repositioning for drugs. It's two concepts to really enhance efficacy and delivery of new drugs in discovery. I've been in pharma, in large pharma for quite some time. I've been been part of discovering a new drug for herpes virus infections in the eye, Foscovir. I've been in that development with a drug for bone metastasis called Cladronate, and I've also been in licensing and alliances with AstraZeneca for quite some time. I've also been in biopharma. I've done combinatorial chemistry. I've gone down a number of areas that have been put into different companies. And now I'm working with an advisory firm in Switzerland, in Zurich, called Notes Advisors. And in that position, I've been having some time to think about how can we do drug discovery leading into good development in a much more efficient way. And that's why I want to share with you. Excuse me. First, I would like to, these are three elements of my presentations. I think that in discovery, my lead terms in discovery is to improve strategies and tools in order to make the better drugs that we all want. So the first part is technological and strategic thoughts, and then we have the repositioning of failed drugs. That means in clinical trials. And if you start with the presentation, saying that often the diseases we're treating are often very complicated biologically, and drugs that we use have several effects. They are so-called pleiotropic. They are usually inadequate precision in determining the target drug interaction. That is just the world we've been living so far, and my goal is to make that better. Let's say have a short historical outlook. In the early century of this 1900th century, there were pre-structure activity relationship time. Things like penicillin was discovered just by serendipity by smart people. In the mid 1900, 1970 and 80, there were SARS got more and more abundant. And for example, antiretroviral drugs for AIDS came through. After that, there was a time where there were people bleeding factory, high throughput screening and combinatorial chemistry, and doing a lot of tests, do a lot of chemicals that would do it, but it didn't really do it. So what's the future pointed to? It's using computerized system, detailed structure activity relationship, and repositioning of drugs that have a long story in development but have failed in phase 2. I put in just one figure, 2021, as to indicate where I think they are. So the source of drugs we had in the early times were bacteria, fungi, or herbal substances found in animals, plants, and man. The combinatorial chemistry that was more specifically done in the 2000s. That's a lot of work. So I think companies like Pfizer showed me at a meeting in 2003. They showed me the new factory for making discoveries. There was a huge robotic setup, and there were thousands and thousands of compounds each day. And with this approach, it was very high tech at that time. It was very elegant, but it didn't produce very many useful drugs, sadly enough. So the pharma companies many pharma companies had problems in sustainability. I worked for AstraZeneca at that time, and you all know that Astra became AstraZeneca in the 2011, I believe. that was a reason for merging portfolios and get around the shortage of new discovered drugs. But nowadays we have the computerized system and are so abundant and they're so powerful that we can increase the amount of data we have, not only the tests, but a lot of different types of data can be put into the system. And I just choose one area called repositioning or repurposing of drugs. You know that every other drug that goes into clinical trials, in phase 2 clinical trials, fail. And when drugs fail, they are lost, they are abandoned. No one wants to touch them anymore. But these drugs have a huge potential because they have preclinical data, have research data, they have wonderful mechanisms of action, and they fail either due to efficacy, lack of efficacy, or with safety, causing safety problems. And that is something that can be addressed in the novel technology is now developed. This is a very, very interesting area. I have founded a company called Actina, dedicated to repositioning of drugs, and I'm about to think about how to make use of the AI opportunity for making new drugs that have a capability to become large useful products. And then I have to mention that Notes Advisors has started an AI effort trying to coordinate people who are interested in this area. How can we use this better? As I said, the repositioning secret is to match drugs and the disease targets. If there's a drug that's developed and it has a wonderful activity for one certain target molecule, it might not be the ideal drug to give to man to treat that disease. Either the mechanism of action is not really a key one, or it can induce safety concerns with the patient. If you take that drug and you look for other diseases, you can find targets that are more meaningful. The good thing with this is that it's a rapid process, and it's a very safe process because the low failure rate that you expect with a repositioning of drugs, meaning a drug that has been through phase 1, safety has been in phase 2, maybe to several hundred patients, you know a lot of the drug. You know how to avoid safety concerns. Focus on the activity. Interestingly enough, repositioned drug contribute 1/4 of the annual revenue of pharma companies' revenues. And there are notably there are top 10 mini blockbusters collectively have sold for almost 13 billion US in year 2015. So I think that repositioning of drug represents a highly valuable source of new model compounds for making drugs in the 2020s. The scientific basis I've touched gently upon, but in summary, the biological targets are often shared in different diseases, meaning that the same target, the same biological molecule can be involved in promoting or responsible for the disease. What diseases, the differences in diseases we see may be caused by the same mechanism. For example, Parkinson and Alzheimer have 48 genes in common related to disease, and that for some drugs may work in both diseases. Another example on the compound side, I mentioned that the drugs are pleiotropic usually, and pleiotropic means that they can affect different of targets. So if, for example, aspirin, this inhibits COX-2 in man at the high concentration, and we get an analgesic effect. But at lower concentrations, it's a COX-1 inhibitor and inhibits platelet inhibition. So you can give patients low doses of aspirin for people with cardiac problems, and you can give high doses if you have pain somewhere. So the secret we have is to understand disease physiology and identify these pleiotropic drugs that act on the right biological target in for each and every disease. And this process is very, very difficult to make in a human person's brain, especially if you do it yourself. It will be greatly enhanced if you use computerized support, AI, to help the researcher, not to do the researcher's work, really to support and help these people find the new and better drugs that we all looking for. If you put that on a time scale, now this is a representation of an entire typical, in my work, my 40 years of experience in this area. It will take about 5-10 years in the preclinical research phase, pre-IND. Preparing for phase 1 will take about 1.5 to 2 years. Phase 1, maybe around 1 to 2 years. Phase 2, 2 years, etc. You add up, there's a lot of years, and that's also translates into a lot of costs. What we can do in repositioning, we can take failed drug out of phase 2, all the data approved for use in man, have a linker study showing that the disease that we are adopting the repositioning drug into is really valid and warrant a new study with that particular drug. We start at phase, that drug will have a short bridging study, we can call it, between former data packages into the new disease description. Then you run the phase 2 study, and after a completed phase 2, a successful one, you will have POC, or proof of concept. And proof of concept in man is really one of the highest value stages in the development process. A biotech company that can produce a positive POC with good statistics has a very good chance to get a collaboration or partnership or sell its drug to a pharma or a very big biotech. What has happened? What factors really accelerate? Why do I claim it can be accelerated? Yes, that's because the SAR data generation has improved. It's been higher and higher resolution technologies that can really picture the target and drug interactions in a positive way. You can more systematically today, out of databases, tell people about the solubility, stability, and other physiological drug properties people have gathered. There are a massive output in publications on that. The use of relevant disease models, both in vitro and in vivo, will increase predictivity to man, which is key in this area. And systematic and complete data collection before cleaning, it's very, very important. That means that you can't take a result out of one publication and compare that with another publication. You have to have comparable parameters, so they are measured in the same sense. It has to be able to compare. If something is toxic, you can't compare two models of toxicity unless you know what you're doing. The AI tool, I'm not a computer guy, but it's possible to handle a huge amount of data and very intriguing, very advanced way. If you are a good programmer, and you can program a machine learning loops into this area. You tell the machine how to learn. I think that we can make a lot of improvements, and that learning has to come from people like us in biotech, supported with the genius people in pharmaceutical programming business. I think this is something that will drive innovative drug discovery. The close collaboration between scientists and software developers is something that is badly lacking sometimes, even in big companies. These types of groups speak differently. The scientists have their word, and the software developers not always understand what the scientist wants, and the scientists can't formulate their desires or their work into terms that can be transferable to the software and environment. And this is something that has to be improved. That's why Notes has been engaged in that. I'm also interested in finding collaboration with this that can help. So if you stay with repositioning, we can find new drugs where we can increase efficacy, reduce safety, have a distribution, excretion, and stability that is really making, let's say, a model drug to a useful drug in man. and we can take that into account. We can make that determination before we really go into the second clinical trial. And we can find novel. I think that we can find really chemical structures by combinations of different chemicals and different effects in many dimensions. We can find really new drugs with better drug properties. And we can, which is financially and very good news for people who are patients, increase the success rate in clinical trials. The success rate of 50% in phase 2 for a new drug is the statistical historical data. We can increase that tremendously. And also shorten the time to approve product, meaning that we can make investors happy. They can get a return on investment within reasonable time instead of this long process usually taken. And we can that will really stimulate every company that has produced a new product, and that product has been sold for good money, and people get better being treated with that, increases the interest of investors to really engage in biotech. That is beneficial for all of us. So the very short development programs with the so-called failed drugs, or the abandoned drugs, is really something that we should be using. Say that every other drug that is in phase 2 fails. You can imagine how many drugs that are available theoretically. With that, I would like to stop and thank you all for listening.
Hello.
Yes, hello.
Hi, Kil. Thank you so much for your brilliant presentation, and thank you so much for being here and adding value. And I'll ask the audience that if they have any questions, they can ask you in the Q&A session or in the chat session. Okay? Can you hear me? Can you hear me?
Yeah.
Hello? Okay. Okay.
I can.
Okay. So thank you so much for being here and adding value. We are looking forward to work again with you in our next conferences. And I'll ask the audience that if they have any questions, they can ask you in the Q&A session or in the chat session. Okay.
Wonderful. It's a pleasure to being part of this exercise, even if it's an e-meeting. It's really fun. So yeah. Thank you very much for
Thank you. Stay safe. Okay, so ladies and gentlemen, our next presenter is Christoph Potempa, who is working as a CEO at Brain Cures. So Dr. Christoph Potempa is a drug hunter dedicated to the prevention and treatment of neurological diseases that affect at least 1 in 6 people worldwide. He holds a Bachelor of Science degree in biochemistry and a PhD degree in neuroscience. After working for a decade in pharma and academia, he established Brain Cures, an award-winning data analytics platform company that leverages a novel biological intelligence approach to shorten drug development processes by at least 4 years and deliver nearly failure-free drug discovery and development. So he is with us. Let's welcome him. And hear me? Can you please unmute yourself?
Yes, can you hear me?
Yeah, I can hear you. How are you?
Good, and you? How can you see me on the camera? I'm not sure.
Yeah, it's good.
Okay, because I see myself I don't see myself in the image.
Okay. Okay, but you're looking good.
Fine. Do you want to wait until 12:15?
No, you can start your presentation now. So
works for you.
Yeah, okay. So
please share my screen then.
Sure. Thank you so much for being here, Christoph, and we're proud to have you here as a keynote speaker and looking forward to have a great session. You can start your presentation by sharing your screen.
Thank you for the invitation. So is the screen being shared?
Yeah, I can see your slides now.
Okay. Okay. Yes.
Yeah, great.
It's fine now.
Yeah. Good luck. Stage is all yours now.
Okay. Thank you. Well, hello. Thank you for the invitation to speak at this conference. I will tell you about brain cures and how we apply biological intelligence to de-risk and accelerate clinical trials. So we are an internationally recognized startup that's been building since 2017, and we are now with most recently in last year we had an article feature in longevity technology describing how we have at least 3 different pathways to better, safer and faster treatments. I will tell you all about it during the presentation. So BrainCure's aim is to deliver precision medicine for brain diseases. You may know that the world is having a big aging population, especially in some parts of the world like Japan. And we don't have cures for many diseases such as ADHD, Alzheimer's disease, depression, epilepsy, pain, Parkinson's disease, schizophrenia. These are all diseases linked to synaptic plasticity. And 1 in 6 people are really affected by brain disease at some point in their life. I think the biggest problem is that there's really the industry average for drug development success rates is 1 in 10. And in Alzheimer's disease, that success rate is just 1 in 25. So that's led to, I guess, some hesitation from pharma companies to work in this area. But fortunately, there is a growing market for brain diseases. And I think just the recent year has shown that there is a, you could say, return into Alzheimer's and some neuro indications. So I think we will have, I think we can achieve brain cures in the next 10 years with progress in technologies. So today, I think it's quite clear that there is a high cost, low success drug discovery era. AstraZeneca did the 5R framework exercise where they look at their pipeline over a decade, and they found that essentially if you don't have a biology linked to your target, you will fail. When you do have a biology attached to your target, a proof of mechanism, you at least get 10 projects, 10% of your projects progressing into the launch. So it's really key to understand the biology behind any disease you're targeting, and especially the neurobiological diseases. So what we need today is novel approaches to drive higher success rates. And these approaches need to be based on quality data that address a specific question. I would say it's more quality than quantity. And what we've done at BrainCures is we've generated a data set that has allowed us to decipher the biology behind transitions from health to disease during learning and memory. So we have focused on the biology of synaptic plasticity. We at BrainCures believe that this is a window to precision medicine. Synaptic plasticity, for the undoctrinated, is a paradigm of the strengthening or weakening of synapses over time in response to increases or decreases in their activity. This paradigm model has guided the development of current treatments for the listed diseases that affect millions of people worldwide. So Alzheimer's, depression, epilepsy, Parkinson's disease, a number of rare diseases like Fragile X, Dravet, and many others, and schizophrenia, just to mention a few. So today's state of the art is really, we've got a lot of missing biological links, and these lead to inadequate treatments, inadequate biomarkers, lots and lots of preclinical and clinical failures. At BrainCures, we've built a molecular map that can help everyone achieve novel treatments, novel biomarkers, and at a higher success rate. So we call our approach biological intelligence. It's a molecular map-driven approach to eliminate preclinical failure and enable between 60 to 100% clinical trial success rates, and also shorten drug development cycles by at least 4 years. And we do this all without deep learning steps associated with artificial intelligence, which I think a lot of we had a talk from Clay, and then 4P Pharma presented some really interesting work earlier this morning. So typically when we start, I think, drug discovery projects, we have in mind the compound, disease, or other data. In the very common today approach, we do apply artificial intelligence, where we do some deep learning, and that generates new haystacks of data, hypotheses, that then need to be validated. What biological intelligence does is it is able to extract the needles from the haystacks based on the time we've invested to tease out a molecular corporation behind brain biology. This brain biology is defined by 822 targets that we then have been able to classify as more connected, you could say, target genes that we call hubs and masters. And if I was to use a corporate analogy, the targets are the workers, the hubs would be the managers, and the masters would be the directors of brain biology. And the better the higher the corporate status, the higher the precision medicine potential is of the compound that is matched to this. So once we build the engine, we ask ourselves, how can we benchmark the product? And so we have identified, GSK has compiled a data set that has within it 72 gene target clinical outcome pairings. And there were 72 phase 3 clinical failures in that data set. When we applied the hubs filter of the BrainFuse discovery engine, we were able to eliminate 57 of those phase 3 failures. And this we could have done as early as the preclinical decision-making point. And so when we then look at the target types and what are the success rates that our engine could give us on that data set, we are able to at least achieve a 60% success rate from target to medicine. And you can see that some targets like the growth factors and cytokines, our engine was able to eliminate all of those failures, with the GPCRs having a rate of over 80%, and transmembrane receptors. So all very common targets used in any of the drug indications. So it's really key to know that the processes we apply de-risk and shorten drug development by at least 4 years. What this means for you is that you cut off at least, most of the preclinical work and those failures associated there, and we are able to essentially provide, I think we feel, failure-free drug discovery. And the savings, I think, are quite huge, as this chart shows here. So I will, I think, show some examples of how the BrainFuse discovery engine works. And we have at least 3 pathways. So one of the pathways is we start with genes and go to treatments. And that involves taking a gene and then identifying what drug it pairs with, and then prioritizing it through our BrainFuse engine, and then doing some, you could say, knowledge searching within PubMed to identify essentially how validated a compound is. And then that guides prospective validation studies that can lead to treatments and biomarkers. I think a very unique part of our approach that the BrainFuse discovery enables comes from involves a pathway from drugs to treatments, where we leverage the data that's in the Broad Institute connectivity map, the L1000, and the signatures that we have in the BrainFuse discovery engine. And there are 98 genes that are common to the BDE and L1000 that we can use to model how the drug will perform. And then this allows to essentially prioritize drugs based on phenotypic data. And you could say third approach is from patient to treatment, where we would apply a biomarker panel and then personalize the treatment selection. So it's almost like a clinical diagnostic tool or companion tool that allows to select personalized treatment recommendations based on knowledge that would be collected from, say, a blood sample from a patient, and then what our engine would suggest would work for a patient in specific diseases. So I will give a few examples of, so this is a Alzheimer's example where we started from an open targets data set where there were 176 genes that were matched 126 compound pairs. This is all retrospective data. By doing the different levels of filtering that our engine allows, because of the molecular corporation we've deciphered, has allowed us to figure out that our framework could eliminate at least, well, essentially increase the likelihood of progression to phase 3 for AD threefold. And it eliminated, well, at least, well, a lot of projects. So the initial set had 717 projects we would have recommended. Only 46 of those as projects that had a chance to succeed with the right selection of patients. And so we can do that kind of, you could say, disease-specific benchmarkings for most diseases as the clients are interested in. And then I think it's very key for me to demonstrate how the virtual phenotypic screen powered by the BrainQ's discovery engine works. So from the drug gene pairs that we identified for Alzheimer's disease, only 55 of the 80 drugs had L1000 molecular data. We then essentially generated this S-curve of the molecular signature based on data that's in L1000, the BDE, to then classify drugs that have a better score than the current drugs used on the market for Alzheimer's. So that's memantine and galantamine. We have identified 20 unique drugs that may outperform current treatments, and here they are listed here. And we have, well, anonymized the drugs here, showing what type of modulator they are in terms of our BrainQ's discovery engine, and also how many clinical trial link publications there are to the between the drug and the engine. So we show here that drug X, which is listed here and has 35 clinical trial link publications. One of those studies will show that essentially it can slow down progression of Alzheimer's disease in patients with moderately severe impairment. There was a study in 171 patients, and this drug has essentially potential to, with further validation studies, to be taken forward and become a real, potentially drug that makes a difference for AD patients. And that could be done obviously by either using pharmacophores of this drug. This would then depend on the marketing strategy of the companies interested in this drug. And then there are other opportunities that are listed here. So here is really a picture that I will then decompose, you could say almost one by one, of how the screening works together with, you could say, further knowledge-based validation. So we've got a lot of things. So we first typically, and this is an epilepsy project where we started with the client saying, well, we're interested in epilepsy. So we said, well, let's look at in open targets, what are the current drugs and gene pairs that are being worked on. Running these pairs through our engine, we're able to classify 20 or prioritize 20 of the 207 drugs because they were linked to 51 of the genes were linked to 51 of their target pairs. We then went on to the literature to discover the novelty of the compounds. And we did that, essentially, we have defined what we call a clinical novelty index. That's based on essentially how many clinical and preclinical publications there are in relation to the compound and disease of interest. And here that was epilepsy. So, there's about 150,000 epilepsy publications out there, and we're able to identify that these, that there were several, low novelty, low risk proposals, a number of mid-novelty, moderate risk that needed, say, further clinical evidence, and then some projects that carried, you could say, higher novelty but higher risk because they needed both preclinical and clinical evidence that's specific to epilepsy. So they had evidence for synaptic plasticity but not epilepsy. And here we show that for epilepsy there are 3 known drugs, and you could say the higher the clinical novelty index, the less prior knowledge there is. So then it depends what the goal of the company is, which for the, you could say, final project selection. We then did a refinement of selecting, of looking for biomarkers, and we did that using the Genomics England panel app, where we then were able to classify these biomarkers down from, so the green light biomarkers, there's 415 of those. We were able to prioritize key biomarker genes down to 48. And then we also found 31 of the 51 targets have clinical biomarkers pairs. And all this work really allows for better prospective experimental R&D that can lead to novel biology and data-powered treatments for epilepsy. So, as I said, that was just a very high-level view of how the process works. And this is more specific breaking down of how we would go, for example, if you gave us any specific disease gene list. And so for one client, asked us to look at 10 epilepsy genes, and we prioritize them in our engine. We did a clinical novelty assessment, network connectivity assessment, and biomarker literature assessment. We then essentially classified these genes, and it's interesting on SNCA1 and SNCA2, which are quite well-known targets, came out either had quite high scores, and we were able to identify that 5 of the genes that the client provided us require clinical validation, and we have identified 381 epilepsy biomarker publications that are relevant to these 10 genes. And so yes, this is very, say, one angle of how the process works. We then did the phenotypic screen where we found 67 epilepsy drugs. 34 of them had the L1000 molecular signatures, which allowed us to essentially then generate profiles of these compounds compared to existing drugs, carbamazepine and levetiracetam. And we essentially here prioritize drugs that fall into the categories of, so either sort of more excitatory model or inhibitory model for epilepsy treatment. And these drugs, each of them has the potential to open novel avenues to biology and data-driven precision medicine in epilepsy. So then, this is one of the drugs selected by the phenotypic screen. It has a good safety and efficacy profile in 8-diazepam-resistant epilepsy patients. So these are obviously all drugs that are, you could say, nearly phase 2A ready, a number that are identified in this virtual phenotypic screens by us. And this is how we could say can personalize, help in the choice of biomarker selection, patient selection strategy. So as I mentioned, there's a resource, the Genomics England has generated, you could say, panels for many diseases. And in epilepsy, there's 568 epilepsy genes, of which the green are the most highly clinical ready biomarkers genes, while amber has, you could say, some evidence and the red lighted biomarker genes have little or no evidence. And our engine was essentially able to highlight that 6 of the 45 little evidence genes could be very interesting. 7 of the 108 moderately ready for clinical use in epilepsy could be prioritized. And then 48 of the 415 that would have very high usage. And some examples of these high clinical readiness biomarker genes include the BRF V600E mutation and the KCNJ11 mutation that allows for, you could say, some precision or specific patient population selection and drug matching. So in summary, we help our clients de-risk and accelerate drug discovery from almost any starting point that you can imagine. So compound disease, other data. We apply at least 3 different approaches. So the BrainCure's discovery engine, we then have been able to have a customized engine that is the BDE 100 virtual phenotypic screening that's powered by 98 genes common to L1000 BDE. And we're able to build bespoke discovery engines for our clients to help you identify targets and compounds with shorter time to market, help you unlock hidden value through repurposing and apply biomarkers for improved clinical outcomes. Today, we're at the stage where we are looking for investment into the data analytics or our own pipeline through partnerships, either through internal or strategic R&D. Our business model and path to market is essentially typically starts with a pilot study with a fee for service that then develops on into a strategic partnership that defines specific license fees and royalties. And in a few years down the road, we expect to have our own pipeline that will allow us to scale up into even a bigger territory company. So I'm very privileged to be supported by very experienced marketing and communication and business development partnership team. So Jean-Françoise Williamson, she has over 15 years of experience as a biotech facilitator across sectors. And Peter Ball has over 30 years of experience in bringing new products to new markets. And one of our advisors is Professor Jacques Mallet, who's a distinguished scientist in neuroscience and gene therapy in the 1960s. And I have over 15 years of experience in decoding biology from data in both academia and pharma. So to wrap this up, the presentation, we are looking forward to work with everyone to deliver precision medicine based on biological intelligence through partnering opportunities for drug discovery, partnering opportunities for biomarker panel development, or a partnership or investment into the BrainCure's platform scale-up. So thank you for your attention, and I'll be happy to take any questions now or at a later time point.
Hi, Christoph. Can you hear me?
Yes, I can hear you.
Okay, great. So thank you so much for your informative and very interesting presentation. And I'll ask the audience that if they have any question, they can ask you in the chat or in the QA session.
Okay, perfect. Thank you.
Thank you so much for being here.
Thank you.
Stay safe. Okay, so.
Bye.
So ladies and gentlemen, our next presenter is Dror Heredis. He is a CEO at VPL Therapeutics. So let's listen to his presentation now.
Everybody. In the next 25 minutes or so, I will share with you some data from our company, VPL Therapeutics, and our lead compound, VP111, which is a transformative gene therapy with a potential to change the treatment paradigm for solid tumors. But let's go to VB111, which is a gene therapy program in solid tumor, which is addressing a major unmet need. And as you will see in the next 10 or 15 minutes, this is a cutting-edge technology on one hand, but very safe and efficient and convenient to the patients and the doctors on the other hand. So what is VB111? So VB111 is actually an information that we give to the patient that turn into the specific drug or protein that do what we want exactly where and when we need it. And in this way, we're imitating nature in three ways. We're using an adeno5 virus, which is a replication deficient virus, in a very similar to what people are doing today to have a vaccine against COVID-19. We're taking this adeno5 just as a vehicle, as a vector to direct the gene of interest to the body. And the way we do it, we take out the gene that enables the virus to replicate. So now it's non-infectious in any way. And instead of it, we put in a very interesting piece of information. We put in a piece of DNA that has both an element of control, a promoter, which everything behind it is expressed exclusively in angiogenic or new blood vessels, which in adult life we have almost exclusively in tumors. And behind it, we put a death receptor, actually a chimeric gene that built from two pieces, which is a death receptor, that every cell that will express it will die. Why do we have death receptors in our body? That's how we keep so young, because we replace old cells, old molecules, and that's the way we refresh ourselves. Unfortunately, in tumors, we don't have this process of apoptosis, but we trick the tumor in the following way. We give this drug, the adenovirus go all over the body and put the transgene that I just described to you in many different cells. But because of this very unique piece of DNA, the promoter that you see here in purple, it's going to be expressed only if we build now new blood vessels. And we don't build new blood vessels in adult life, almost never, unless we have a tumor, which is like an organ that needs blood vessels. The moment that the tumor is trying to grow, it's taking normal endothelial blood vessel cells and make them angiogenic to grow new blood vessels. At that moment, the frame of this promoter is readable by these cells, and the transgene is expressed. The transgene is built from FAS as a death receptor intracellularly, but the receptor outside of the cell, it's tumor necrosis factor receptor 1, which, as you can hear from the name, it's a compound that is always exist in tumors to enable it to actually grow and build in the normal tissue. So the tumor is actually activate this receptor and kill the new endothelial cells. The tumor will go to take another cell, and the same process will happen again and again and again. Because of this, the tumor will try to grow, but won't have enough blood supply, and we will see a necrosis in the center of the tumor. And of course, eventually the tumor will shrink. But not less important mechanism or part of the mechanism, because we have here a dual mechanism, is actually the point that we are using a virus. And as you know, the immune system is not built to fight tumors because they are basically our cells. They are built to fight viruses, bacteria, and so on and so forth. So the point that we did put a viral genes within the tumor is actually bringing the immune system, especially CD4 and CD8 cells, into the tumors and make what we call the cold tumor that has no immune system to a hot tumor where the immune system is there, start recognizing the tumor and killing it. And by these two mechanisms, we get a major anti-tumor effect or immune response. So let me show you that what I just told you is actually what's really happening in our body. So this is a lady from the phase 3 pivotal trials that we are running now in ovarian cancer, in platinum-resistant ovarian cancer, which mean ladies that had ovarian cancer, got surgery, got chemotherapy, many cycles of chemotherapy, almost everything you can imagine, and now they're coming to us because they failed everything. When we look at this patient, in the beginning, she has no immune cells in her tumor, but CA-125, which is a biomarker in the blood, is actually close to 1,000. The normal level is 35. And she has a big burden of tumors with metastatic lesions in her liver. Each time you see this purple virus, she's getting a dose of VB111 every 8 weeks. And you can already see that she really beat the progression-free survival that's supposed to be 3.5 months. And she was getting this treatment for over a year. After the first dose, we already see inflammatory cells and we see a major response. The CA-125 is lower by more than 50%. And then gradually, it's coming to the normal levels. After the third dose, you can see that there is a lot of inflammatory cells. I told you that the tumor will get necrotic because we don't give them blood supply. As you can see here, the top lesion just disappeared and the lower one became dark, which means fluid, necrotic. There is almost no tumor. And in June, you can hardly see any tumor there. We did biopsy from these lesions. That's part of this protocol of the study. And as you can see here, that's before treatment, a lot of tumor cells, no inflammatory cells. After the first dose, still you can see tumor cells and a lot of necrosis, but a lot of CD8 and CD4 cells within the tumor, which is an exception in ovarian cancer, because ovarian cancer is a cold tumor. We turned it hot. Now, after the third dose, the whole tumor become necrotic, and the only thing you can see is a lot of inflammatory cells. Of course, this can be translated into other cold tumors, and indeed we are doing now another trial together with the NCI, National Cancer Institute in Washington, looking at patients with colon cancer, which is known to be a cold tumor, where there are failure of a chemotherapy and had metastatic lesions all over. And we're giving VB111 to make their tumor hot. And then we combined it with checkpoint inhibitors to activate the immune system. So what do we know about VB111 and how did we start developing it? So the first thing first, we did a phase 1 for safety. And in this phase 1, we started with 6 different cohorts in low dose, single dose of VB111. And then the top one in blue is cohort 7, where we already gave as a single dose 10 to the 13 viral particles, which is a therapeutic dose that we are using. And as you can see already, these are days that these patients that had different type of tumors with a lot of metastatic lesions beyond any therapy, all of a sudden, instead of dying in this cohort, they were keep on living and living and living for years. We're talking hundreds and hundreds days. We then had cohort 8 and 9, where we were giving it in multiple dose, and these patients were alive for many years. So we knew that there is efficacy of VB111 when you get to the right dosing. In parallel to this trial, we started another trial in specific indication in well-differentiated thyroid cancer. And the reason we did it, because we had 2 patients here in this cohort that we had a signal that the drug is working in thyroid cancer. As you can see in red, we gave it as a single medium dose, and yet there was already an effect and the patient were living longer than expected. But when we gave it in 10 to the 13 every 8 weeks, 40% of the patient kept on living for years and years and years. So you can see again a nice dose response. Again, because we had some signals that it's working in the brain from this trial. We went to do a trial in GBM. This time we were very careful because we knew that because we are breaking the blood vessels, we will create edema, and in the brain it can be dangerous. So we started with low dose, and then we increases to 10 to the 13, and eventually it was a single dose because all the doctors noticed the edema, the growth of the tumor, although it's pseudo progression, and actually didn't give a second dose. But then we agreed with the FDA that we will actually start with VB111, prime with it, and if the patient progress, we allow them to go on standard of care, but keep on VB111. Most of the patients went on Avastin because that was a drug of choice at this point. You can see that we double the overall survival in this very difficult tumor. But beside of this, we were getting a call from the doctors in the OBGYN at Harvard asking us to do a trial in platinum-resistant ovarian cancer. And the reason was that they were involved in the phase 1 and they noticed that there is efficacy for the drug. So we started with a low dose because this is the first time we combined it with chemotherapy. And the patients that didn't get the right dose actually survived as expected in this very difficult population, about 6 months. But the patient who got the right dosing of VB111 kept on living with a median half-life of about 500 days. And the one that had an objective response to the drug, actually the median survival was 800 days. So we knew that we have a very strong signal in ovarian cancer, and that's why we decided to go and do a trial that will help us to develop this drug for platinum-resistant ovarian cancer. So from the mechanism of action on what I showed you so far, you can see that this drug is not going to be aimed only for ovarian cancer. It has a potential in lung cancer, in thyroid, in brain cancer, in liver metastasis, and in GI cancer. And I'll share with you more data in the next couple of slides. So where are we right now with VB111? We are running 3 different trials. The most important one is the ovarian cancer oval trial in platinum-resistant ovarian cancer. We're doing it with GOG, which is the American Gynecological Oncology Organization, and we are running it and with Nanocarrier in Japan, and we are running it in the US, Europe, Japan, and Israel. Beside of that, we are doing another trial in colorectal cancer in combination with nivolumab with the National Cancer Institute, and we are running another trial in recurrent GBM, or glioblastoma, the most deadly brain tumor, and this is an investigator initiated by 7 major centers in the United States. Dana-Farber, Mass General, Memorial Sloan Kettering, UCLA, UCSF, University of Texas, and University of Utah. And of course, they are running the trial for us, but the data belong to us in all of these trials. So what is the OVAL trial, which is the most important trial? This is a randomized phase 3 double-blind controlled trial, which is a registration-enabling study in platinum-resistant ovarian cancer. As I told you, we're running it in these different countries with GOG, and it's a 400-patient trial where we basically randomize a patient 1-to-1. Half of the patient will get paclitaxel with placebo, and half of them will get paclitaxel with VB111. The paclitaxel is given every week, but the VB111 is actually giving every 8 weeks. Because we are running such a big trial after a relatively small phase 2 trial, we agreed with the FDA to do a trial within a trial in the beginning of this study. And when we got to about 60 patients that were randomized, 30 in each arms, we were allowed to look at the biomarker response, CA125. And we built it in a way that we won't be unblinded, but the DSMB, of course, the committee was completely unblinded. And they assured us that we have at least 10% absolute difference in response in favor of VB111. And we know now that at least 58% of the patients had a response. It's a known thing that in this late stage, only small number of patients really get a response. So basically, Now we are running the trial when we already know that we are actually meeting one of the secondary endpoint, which is CA-125 response. But this trial is unique because we have two primary endpoints. Each of them will be enough to put the drug on the market, and that's what agreed with the agency. Both progression-free survival and overall survival will be sufficient to put a BLA and put the drug on the market. Of course, PFS is going to be done about a year before we are going to look at the overall survival. Just to show you how the response rate that we were getting with VP-111 is so much higher than what you would expect with chemotherapy alone. So definitely the response rate already telling us that VP-111 is most probably working in this trial. This is a unique drug because all the other drugs that has been approved in ovarian never showed overall survival. All of them showed only progression-free survival. And we hope to repeat what we've seen in the phase 2, which is actually showing an overall survival. In terms of safety, this drug is extremely safe because it was actually not discovered, but designed, as I told you, the vector, the promoter, the gene that we picked. This drug is actually the only significant side effect is flu-like symptoms with fever and chills. We see a little bit of thromboembolic events, which you do see much in tumors, and we didn't see more in with our drug, but eventually, as you know, adenovirus can create a little bit the tendency to wear thromboembolic events. In terms of what's the market that we are aiming to, it's actually multi-billion-dollar market, and therefore we are actually developing a drug that can be a blockbuster and can be a game changer in solid tumors. This is a very unique, sophisticated drug. It's a gene therapy-based drug. Actually, before the COVID-19 pandemic, it was always quite difficult for me to explain how and why we are using a virus and how do we do this as a drug. So because we were so much ahead of our time, we built our own commercial grade facility in Israel with the help with the Ministry of Health. And this facility is working in a commercial grade, already been approved or audited by the Ministry of Health in Israel, by the European QP, and just lately, the FDA authorized the clinical use of VP-111 batches that produced in this facility after a thorough discussion and looking at all the data that we could submit on all the batches and the releasing tests that we are doing. So basically, this facility was already audited by the Israeli authorities and have been QP by the Europeans and looked for the data by the FDA. When are we expecting to see the data? For the progression-free survival in the second half of 2022, a year from now, and for the overall survival a year later. We expect that if the PFS come the way we hope that it will come, we will be able to submit the BLA by March 23 or the first half of 23 and actually put the drug on the market during 2023. So what I showed you so far is what we have at VBL and what is VB111. We're a public biopharma company with our own GMP gene therapy facility. We are well-funded. We are developing a cutting-edge technologies for solid ovarian cancer. We have a phase 3 asset where VB111 is already late in stage in this phase 3, and this can potentially be a blockbuster drug. And we hope that we are on the way to potential commercialization of VB111, first in platinum-resistant ovarian cancer, but later on in many different indications and changing the landscape of treatment of solid tumors. Thank you very much for listening to this talk. Thank you.
Okay, hi, welcome back. I hope you all have enjoyed the presentation of Drawer, and it was really very interesting session. So now our next presenter is Raul Insa. He is the CEO and founder at Som Biotech. And Dr. Raul Insa has been working for more than 25 years in multinational pharmaceutical companies, such as Parker Davis, like now Pfizer, UCB Pharma, Urea Group, and ICTIN. His experience is related to drug development, from drug identification to commercialization, including mainly clinical research and licensing. He was involved in the development of products. So let's welcome Raul. Hi, Raul, can you hear me?
Yes, I can hear you.
Okay, great. So how are you?
Very well, thank you.
That's great. Good afternoon. Thank you so much for joining me and for adding value in this conference. And you can start your presentation now by sharing your screen.
Okay, great. Thank you very much.
Thank you. Stage is all yours now. Good luck.
Yes, that's it. Can you see my screen?
Yeah, I can say it.
Okay, that's perfect.
Okay, great.
So here we are. So first of all, good afternoon, everybody. Thanks for the invitation to this interesting Drug Discovery Summit conference, where we can discuss what to do to improve drug discovery at all levels. I will talk about our experience by using artificial intelligence solutions in drug discovery applied mainly to orphan indications, and well, put some examples on what Som Biotech has been doing in the last 10 years on this. So first of all, I would like to draw just some few numbers that everybody knows for sure about drug discovery and the amount of drugs that are there available being discovered in the past for different indications. It's around 12,000 molecules that have been used, are used, or were used in the past for different indications. Okay, which is very, very few amount of molecules that can be druggable, okay, or which are drug-like. Okay, according to some publications, there are about 10 to 33 or 10 to 60 potential drug-like molecules that can be discovered for different diseases. But we're still working with very few of those. Okay, small molecule drugs that are available there in the market are about 10,000, but the potential is extremely high. If we do a very simple comparison, okay, there are 10 to 21 water molecules in a drop of water, but in the overall seas and oceans of the Earth, it's about 10 to 46 molecules of water there. Okay, so there are many possibilities that the only way to address this complexity is potentially by using artificial intelligence that, as you know, can work for a great amount of data in order to choose that is more interesting for us to work with, because if not, it's impossible. And also we combine that with drug rediscovery. We call it a rediscovery, but you can call it repurposing, okay, or switching, or drug profiling, okay, which means to take the drugs that are available and to see if those drugs can be applied to other indications. Then it makes potentially it makes more easy, or at least less expensive and less risky. When we compare, this is my next slide, when we compare the cost or budget that you have to invest in a new chemical entity, which is right today close to $2 billion, and you see how a repurposed drug that already exists in the market, and you know about the safety, the quality of the drug, but not, of course, the new indication, which may be 50 million, or even if it's larger, 100 or 200, you are really reducing a lot the costs in the development of that drug. The time also can be reduced quite a lot in the terms of time to market, okay, because it's from 10 to 70 years for a new chemical entity, but it can be from 3 to 5 years in a specific indication. If it's orphan, it can be even closer to that date, to that data. So there are some benefits of drug repurposing. Okay, not only benefits for the patients because they may have a new treatment with a lower cost. Benefits for the healthcare system because it can be approved earlier and the expensive or the cost for the system may be lower. For the industry in terms of times to discover costs and risks. Okay, for the authorities to approve drugs also as well, very interesting. Okay, and worldwide, it's about 25% of the annual pharma revenues. Okay, 1/4 of the revenues of the pharma companies worldwide are repurposed drugs. Okay, drugs that are reformulated or reindicated in new indications. So it's part of the life cycle or life cycle management, okay, of a specific medicine or drug. There are many examples. Okay, this is just a table, okay, that probably you cannot see properly, but it's a table of drugs that were discovered for a specific indication. Later on, they had new indications that in some of the cases became a blockbuster. And this is the case because, as you know, and you have seen during the morning, drugs are very promiscuous. Okay, if we look into drug bank database, we can see that there are about a mean of 3 targets or 3 places where the drug acts, okay, per drug on that database. Okay, so it means that about from 2000 approved drugs, okay, that we have seen in this database, most of them do have different mechanism of actions. Okay, so drugs are very much promiscuous, and the interesting thing is to find these new uses of those drugs that can make affordable the development of that specific drug. So the approaches or ways to do this are very different. Okay, you can use in vitro studies, so to try to do high throughput screening and to test everything that is available to see if you can find something. You can use a clinical approach, which is usually based on adverse events, or I would say unexpected events. They don't have to be adverse. You know, you may expect something which is not expected in advance. For instance, sexual dysfunction in the case of Viagra. Okay, and you see that event, which is not really adverse. It can be used to repurpose the drug for another indication. Or you can use literature, okay, many papers, many patents, many ways to try to guess where a new activity of a drug may happen. Also, you have in vivo studies, okay, you can do phenotyping screening of the drugs in order to identify new activities. But there are other ways, as the in silico, to use computers, which are usually cheaper and faster, by using 2 dimensions or 3 dimensions QSARs or toxicology studies of pharmacodynamics, structural data, and other approaches in order to identify that potential activity of a database of drugs. Okay, this is usually what we do in the company I founded, Sun Biotech. Okay, and we base that on an artificial intelligence platform. But you can use also other in silico approaches like big data approaches or machine learning or other things. Okay, how popular is the use of artificial intelligence in drug discovery? Okay, you can see in this chart that it's increasing during the years. It's increasing quite a lot. It's expected that in 2024, the use of artificial intelligence applications with drug discovery, which is the green part of the bar, will increase at least the double of what is happening by today. Okay, And as you know, because of my previous slide and talks, it's because it's faster, cheaper, and of course, it has to be predictable. Not all the times is as predictable as we would like. If we look into artificial intelligence news in Google in the last years, the trend has increased quite a lot. And today you can find a lot of news and I would say interesting space in what is artificial intelligence. Even though it's true that many of the technologies that we consider artificial intelligence are not really driven by artificial intelligence. Okay, so it's very fashion word, but we have to be very cautious on what we consider as intelligence in the artificial world of the computer systems. Coming back to some biotech, okay, as I mentioned before, we're leveraging our drug discovery in a pioneering technology, which we call some artificial intelligence pro. And why are we claiming that this is a very good solution, even it's not the solution for everything, okay? Because in our hands, in 29% of the projects we have identified to this technology positive hits in vitro, okay, when we validate those findings of the computer in the lab in vitro. In 12% of the cases, we have arrived to phase 2 positive data in humans, as you will see later on in our pipeline. Okay, this means that we started in 2010, 16 projects in orphan indications, and 2 programs have arrived to phase 2 with positive data in patients. Okay, that's where the 12% comes from, which is a very, very good success rate. You know that the success rate for new chemical entities is about 1 to 2 products, up to 10,000 products that are synthesized in the medicinal chemistry part at the beginning, okay, that will arrive to the market. But we have been quite successful with our technology. This technology is around 3 times more predictive than other technologies we compared in the past. It's based in molecular fields, and the beauty is that it identifies non-structural analogs of a given compound. So we can identify chemical structures which are absolutely different to what is known to have activity in a disease or a specific mechanism of action, and that can be, of course, protected through a patent. It can be applied to any therapeutic area. As far as new chemical as chemical entities, we are talking about small molecules, okay, not large biologics or big peptides. And we have been using, as I said, since the last since we started the company in 2010, in different approaches to save time, cost, and to lower the risk of the development of drugs. The technology, this artificial intelligence technology, is based in genetic algorithm. That's a branch, as you know, of artificial intelligence applications that modifies the molecular fields around a drug in order to interact with other molecular fields of a database of drugs. And by doing these interactions at least 2.5 million times, it learns which is the best molecular configuration that fits with a specific molecule, and it identifies these molecules from this database according to this genetic algorithm. So it does not need initial information, really much initial information to start a search, only a chemical structure. It can explore multiple parameters, and it's quite fast. Usually within 2 days, we have the results of the machine, and of course, later on, it's the human brain that has to work on that results to see if the candidates can be druggable or not. Our typical approach to drug development is quite a standard. Okay, we use an orphan indication where we want to work. We use our technology, some artificial intelligence pro, which is, as I mentioned, based on molecular fields, and it's a ligand-based technology. We identify some candidates, we validate them in vitro or ex vivo or in animal models, depends on the specific disease, and then we file a patent. We asked for orphan drug designation. That's one of the beauties of working in orphan disease, as you know. And we proceed with the clinical development plan to start phase 2 proof of concept as soon as possible. Okay, that's the idea, to reach human beings as soon as possible, to have the proof of concept that it's really working in humans. Because there are many drugs that do work in animals or in vitro, but they are not working as we expect in humans. So we want to reach patients as soon as possible. That's why it's beauty to work with repurposing drugs as well. And we push the development until the drug is approved in Europe and United States, or we are licensed that asset to another company. The most important thing working in repurposing drugs is to have a strong market protection. Okay, as the drug is there in the market or has been in the market for some years, or maybe in the market in the future, it's very important to protect that commercial risk, because at the end of the day, we are doing business, so we want to recover the investment, and investors do want to recover the investment that they put in any company. So we usually file a patent for this new use or application. We work on new composition of matter patents for new formulations of the drug, or even new strengths on the drug for a new indication. We ask for orphan drug designation to have exclusivity, regulatory exclusivity in the market from 7 to 10 years, depending on the territory. And because of the orphan space, we try to look for fast track designation, priority or accelerated approval, and so on. As I said, the technology can be applied to different therapeutic areas. Okay, as there is, if there is a small molecule or a small peptide that has a known activity in humans on that disease, we can use the technology directly. But we decided to focus in orphan indications as a company, not to be dispersed, to be more focused. And also because in the orphan space, as you probably know, there are around 7,000 diseases, and almost 4,000 of them do not have any treatment. So it's a therapeutic area, or part of the more than therapeutic area, a part of the diseases that is growing. In terms of market value, it's growing more than 10% annually. Its market value is around 140 billion. And 20% of the orphan drugs are repurposed drugs. Okay, so it's not so unusual to identify repurposed drugs in the orphan space. We also focused on CNS mainly, because there are a lot of conditions that affect the CNS, and the market is around 90 billion orphan CNS conditions. Okay, last a couple of years ago. So there was a lot of room to innovate and to develop drugs there. So by applying this strategy of repurposing drugs to an artificial intelligence technology in orphan diseases mainly, we have reached this pipeline as per today. We out-license 2 drugs for the treatment of one TTR amyloidosis and the other one for glioma and other types of cancers. So the first asset was out-licensed after phase 2A in a population of 20 patients with TTR amyloidosis. We saw that we could stabilize the TTR protein in blood, and after that we out-licensed worldwide rights to an American company based in New York. The second one for treatment of glioma, it was not really a repurposed drug. It was a new chemical entity that we discovered from a database of chemical structures, which means that this drug was never developed or never used in humans. But we saw a nice activity in glioma after our in vitro studies. Because it was very risky, we decided not to develop it by ourselves. But we were lucky to have licensed it to a Singapore company called UCell Therapeutics, who is in charge of developing the drug for cancer. Right now in the company, we have 3 assets under clinical development. The first one, SOM-3355, is a drug that was used in the past for hypertension. It's a beta blocker. It's still used for hypertension in some Asian countries. But we discovered a new activity as a vesicular monoamine transporter 2 inhibitor, a VMAT2 inhibitor for the treatment of chorea in Huntington patients. It's a symptomatic treatment, but almost all Huntington patients do develop chorea. and Korea is very life-threatening for them. So it's and they need a good treatment because there are only 2 drugs approved in the United States, 1 in Europe, and both drugs, which are tetrabenazine and deutetrabenazine, do have a black box because very serious adverse events. So our drug has been used for hypertensive patients in thousands of them in the past. It's very safe. It has not these safety concerns. And the efficacy is very similar because we did a phase 2A in 4 hospitals, 32 patients with very good results. We asked for orphan drug designation and pre-IND in the FDA. And right now we are running our phase 2B dose finding study in Europe. So we expect to have the results by the end of next year and continue with the phase 3 in order to have approval of the product in Europe and US by ourselves. The second drug, SOM 3366, is a derivative of SOM 3355. It's an enantiomer of the previous one. And because of the characteristics of this enantiomer in terms of the promiscuity, as we mentioned at the beginning, our intention is to focus this drug on tardive dyskinesia, another indication which is driven by the same receptor, VMAT2, but the specifications are slightly different. And it might be also useful for other indications we are exploring right now. So we're going to start a phase 1 because it's an enantiomer, and we have to qualify better the pharmacokinetics of the drug as soon as possible. The third drug, SOM 1311, is a very interesting one because this is for the treatment of phenylketonuria, PKU. There are only a couple of drugs in the market that address a specific mutation of the disease. We have identified one product and there's another one very similar to this one, but for the different indications that are in the same program. And we can start phase 2A patients in Europe probably by the beginning of the year. So it's a very, that's why by using artificial intelligence and repurposing, you can jump from discovering a drug and analyzing the business case of that drug in terms of commercial sense and drugability, and potentially jump into the clinics in 1 year, okay, which is extremely fast, I would say. We also discovered one drug for 3 drugs, but one was very interesting for the treatment of COVID-19 as a protease inhibitor. Okay, but right now everybody is more focused on the vaccines and the prevention of the disease than in the treatment of the disease. So we decided strategically to focus on our orphan pipeline instead of the COVID-19. Also, we have some candidates in different orphan indications like Duchenne muscular dystrophy, Niemann-Pick type C, a specific type of Parkinson disease linked to alpha-synuclein mechanism of action, also Tay-Sachs and Sandhoff. So right now we are choosing which of these promising drugs, because of the in vitro models, are the best to be druggable in order to push them into the development pipeline, into the clinics. And in parallel to this pipeline, we have almost around 20 more programs which are at discovery stage. Discovery for us is the in silico part and in vitro part. Okay, as soon as we have in vitro results, we decide to select the candidate or not. So our technology is very much, I would say, promising. Okay, our team is quite small. We are right now around 15 people. Okay, worldwide, and this 15 people is running all this pipeline, and also, of course, the technology and doing some collaborations with companies. Sorry. So let me just put a couple of examples of how we have developed the 2 main programs, the TTR amyloidosis and the Huntington program, and you will see how we can apply this artificial intelligence technology to repurposing drugs in orphan indications. So this program started in 2011. At that time, there was only one drug called tafamidis from Pfizer, which was under development by an American company called FoldRx, and it was in phase 3 Pfizer decided to buy this company, paid $400 million, something like that. Got the rights for the families, developed it, and right now it's already in the market. But there was nothing in the market at that time for the treatment of TTR amyloidosis. So we took the families as our reference compound. Okay, it means that the technology, or artificial intelligence technology, takes into account the families, builds the molecular fields around the chemical structure of the families, does all the million of interactions that it has to do to identify in a database of around 10,000 compounds, which are the compounds that have been commercialized, are commercialized, or were in the market at some point, which are the best compound that matches those molecular fields in order to identify some better activity or similar activity. And the system, some artificial intelligence pro, discovered a chemical structure called tolcapone, okay, that you can see in the slide. Tolcapone is a drug that was approved a long time ago for the treatment of Parkinson's disease. It's a catechol O-methyltransferase inhibitor. Its mechanism of action is to inhibit this enzyme in the liver in order to allow levodopa to reach the brain in Parkinson's patients. It was launched in 1997, so it's an older drug already in the market for a Parkinson indication, along with levodopa. The chemical structure, as you can see, is very different, so that's why it's nice to think out of the box and use artificial intelligence technology or type of in silico technology, because you can be able to identify non-chemical structures which are non-analogs of a given structure that you know that is working. So first of all, we did our in vitro studies comparing to the families. We discovered very soon that some O226, okay, which is tolcapone, is 4 times more active in binding to the protein and stabilizing the protein than the families. And we did our phase 2A study in these 20 patients, and we discovered that 2 dosages that we could stabilize at 100% the TTR in blood. So after those initial studies in this proof of concept study, we outlicensed it to the American company, and they are developing a new formulation, started phase 2B, specific indications of the TTR world, and so on. Second example is, as I mentioned before, our drug for the treatment of chorea in Huntington's disease. Okay, in this particular case, we took as a reference compound tetrabenazine. Okay, tetrabenazine is a generic drug in US and in Europe, which is linked to a black box in the leaflet because of serious adverse events, mainly suicide, depression, Parkinsonism, and neuroleptic syndrome, which is little. So it's very quite unsafe drug, but it's the only thing available in the market at the time. So physicians, neurologists do have to use it if the chorea is associated with a behavior of the patient that really invalidates his life and the way he eats. So we took tetrabenazine, we introduced it in our system. Our system did the same as before, looked at the molecular fields, compared those molecular fields through different iterations with the database. And we discovered that bevantolol, a beta blocker, discovered in US by an American company, later on licensed in Europe, and later on to Japan. So that's why it's only in ASEAN markets. This beta blocker with a totally different chemical structure than tetrabenazine has very similar activity. As you can see in this graph, the in vitro assays showed that the activity as a VMAT2 inhibitor IC50 is very similar to tetrabenazine. So because of that, we jumped, and because of the safety of the drug, we jumped directly into a phase 2A clinical trial in 32 patients, where we reached our goal. We reached our positive, okay, the primary endpoint, which was to reach at least around 57% of patients that had a response more than 2 in the total maximum chorea scale. Okay, and we used 2 dosages, and the highest dosage was statistically significant to the previous one than to placebo. placebo. So that's why we decided to do a phase 2B dose finding study. Okay, we looked for FDA pre-IND or for drug designation and everything in order to continue with the development of the drug. Another example, which is not orphan that I mentioned before, is our COVID-19 program. Our COVID-19 program is also very interesting because last year, around January, February of last year, when the pandemic situation was starting, I would say, in Asia, and later on came to Europe and US, we decided to use our technology in order to try to help. The Chinese guys had identified a COVID-19 protease inhibitor, which we used as a reference compound. This inhibitor could not be used in humans because of safety concerns. So we said, we decided, let's use our technology and see if we can identify another compound that is safe and can be used as a protease inhibitor as a first-line treatment of patients that arrive to hospital. The technology identified different compounds, but in vitro, we validated them and we identified 3. And this was done in Korea, the Women's Hospital in South Korea. And we identified everaciclin, dexacertib, and cynarin. We filed a patent on them, and we choose eravacycline as the best drug to be repurposed because this drug is available right now in the United States market for treatment of gram-negative infections in hospitals. It's intravenous administration. It concentrates very well in the lung and potentially crosses the blood-brain barrier. So it was a good candidate to treat COVID that, as you know, was or is still a lung infection or affecting more the lungs when the patients are hospitalized. So we did some in vitro experiments that validated the activity of the drug. And right now, because of a collaboration with the Korean government, we are working on an animal model to validate the activity of everaciclin as a protease inhibitor. We asked for pre-IND FDA. We have the protocol proof of concept in humans in place. And well, the thing is that we have to partner this program, okay, because we don't want to focus in COVID-19. And it's also from the business point of view, it's very difficult to assess whether in the future an intravenous protease inhibitor will be required or not if everybody is vaccinated and we don't have any more COVID. But well, this is difficult to address. So right now we are having it in an animal models to verify the activity of the drug. But again, it's an example of how our technology works in order to bring drugs that already exist for another indication. And this is all about my talk. Okay, I don't know if there are questions right now or we have to wait until the end of the presentation.
Hi, Raul.
Hi.
Hi, Raul. Thank you so much for your wonderful presentation. And we are really honored to have you as a keynote speaker. And thank you for adding value in our conference. So I'll ask the audience that if they have any question, they can ask you in the QA session or in the chat session. Thank you so much. Can you hear me?
Hello?
Yeah. Hi, can you hear me?
Yes, it's Eric.
Okay. Hi, Eric. I was just, hi, Raul, can you hear me?
Yes.
Hi. Okay. Okay, great. So Raul, thank you so much for being here and adding value in our conference. And we are looking forward to work again with you. I'll ask the audience that if they have any questions, they can ask you in the QA session or in the chat session. Okay. So thank you so much. Have a great day. Stay safe. Okay, so ladies and gentlemen, now our next presenter is Eric Keliova. He is here with us. And hi, Eric, how are you?
Fine, thank you. Thanks to have invited me, Sarina, to this meeting. Hello, everybody.
Thank you so much for joining us, and we are really happy to have you here. And Eric, ladies and gentlemen, this is Eric Iliova. He is the president and CEO from PDC Line Pharma. He is as well board member of the biotechnology company Bioxort, Hairclon, Verticell, and InnoBiochips. He has raised more than $80 million euros over the course of his career, and has had numerous successes in the scale and the initial public offering of biotechnology companies. He is co-inventor of the first GMP approved mobile manufacturing unit for cell therapy. Eric is a co-founder of 4 biotechnology called Myosix. Okay, so Eric, thank you so much for being here, and you can start your presentation now by sharing your screen.
Yes, I will share my screen right away.
Okay, the stage is all yours now. Good luck.
So I believe you can see me.
Yes, I can see you and
Okay.
your slides as well.
Great. So I'm going to present you over the last 30 minutes the activity of PDC Line Pharma. So we are a biotech company, European biotech company involved in cancer vaccine based on a very interesting technology, which is an off-the-shelf antigen presenting cell line. So the company is a spin-off from the French Blood Bank. In fact, the scientific founder of the company, Joël Plumat, when he was leading academic research lab associated to the French Blood Bank, has identified in the blood of a patient a very rare leukemia, a leukemia affecting the plasmacytoid dendritic part of the cells. You know, the dendritic cells are the cells that are educating, priming, and expanding CD8 cytotoxic T cells, the T lymphocyte that kill tumor or destroy viruses. And in the majority of his dendritic cells are myeloid, and a tiny part, few percent, are plasmacytoid. And this, the leukemia was affecting these small portions, and these cells have very interesting biological property. Based on the blood of his patients, he developed a master cell bank approved for clinical use, fully characterized, and that we are still using today as a vaccinal platform. We just have to stick GMP peptide representative of tumor antigens on the HLA present in the membrane of the cells to address any kind of tumor. So today we're a team of 25 people, including seasoned executive from biotech and pharma. In terms of funding, we get close to 35 million euros of funding. We will announce soon an additional B2 round of funding. Our proprietary PDC line has 3 key advantages. So first of it is a cell line, so it's immortalized, very easy to expand and grow in culture. You will see in bioreactor without any cap serum. Second element, it's a plasmacytoid dendritic cell, so provide very interesting biological property and capacity to expand CD8 T cells, cytotoxic CD8 T cells, and I will show you some benchmark study. And last is the versatility of the platform. In fact, as I said, we just have to load the cell line with GMP peptide representative of the antigens, so we can potentially address any kind of solid or liquid tumor. So it's broadly applicable to any kind of cancer. We are clinical stage, so we have already completed an academic feasibility study in melanoma, and we have a phase 2, 1-2 ongoing in non-small cell lung cancer. So just a summary of the pipeline of the company. As you can see here, we have 3 key components. The first one, PDC-MEL, that is the academic clinical trial on melanoma with 4 shared tumor antigens. The phase 1, this study has been completed. The result has been published and is available in the website of the company, the scientific paper. The second key component is the PDC lung for non-small cell lung cancer with 6 shared tumor antigens. The phase 2, 1-2 is ongoing. And we have at preclinical stage PDC neo, which are shared neoantigens, in fact, frameshift related neoantigens that we could use as to address new indications. Important to mention that we have signed already a first licensing deal in early 2019 with LG Chem. So LG is quite known, at least in Europe and US, for the electronic and the flat screen, etc. But it's as well the 10th chemical company in the world, and they have a growing life science divisions. And we have signed a deal with them, and they have the right for the lung cancer vaccine with an exclusive license in South Korea and options for the rest of Asia. You see the total deal value below. It's $123 million plus significant royalties on net sales in Asia. The management team is one of the key assets of the company. You see here a snapshot of the bio of the management team. Myself, I've co-founded already several biotech companies in cell and gene therapy, sold one of them to Genzyme. The medical director, Shanna DeBroy, come from GSK Vaccine. She led the cancer vaccine clinical trials for lung and melanoma within GSK. Claude Edry is in charge of the CMC and he has a long experience in cell therapy. He's as well a former GSK vaccine executive. The scientific founder, Joël Plumin, which is that was leading the academic lab, joined full-time the company. And we have Laurent Lévy, the CFO, that was as well at the origin with Joël of the company. Important to mention that in the board, we are very senior and competent people. We have as a president, Monder Majoubi, who is the former senior VP of Genentech Oncology and AstraZeneca Oncology. Jean-Paul Prils, that is the former global R&D director of GSK Vaccine, or Alain Herrera, that was the former business unit head of oncology of Sanofi, plus of course representative of the investor. We have a mix of Belgium and Korean investor within the company, like, for example, a Korean investment partner, which is one of the key investors in the company. But we have as well François Fontaine from the Belgium Sovereign Fund, which is one of the leading investors within the organizations and the share structure of the company. So in terms of achievement, the company made significant achievement over the last 2 years. We get GMP accreditations by the Belgium Agency for the QC and release activity of the cancer vaccine in lung in January 2019. In March, we closed the licensing deal with LG Chem I mentioned earlier. In June, we get the authorizations to launch the clinical trial in Belgium and France. In December, we closed a 20 million B round of financing led by a Korean investment partner. In February, we announced the administration of the first patients with the PDC lung low dose. In April, we announced the publications of the international patent application related to modifications of the cell line. I will come back to that later. In June, we get the new president of the board, Monder Majoubi, joining the company. In September, we announced the administrations of the patient with high dose of the cancer vaccine, get authorizations in November. And last but not least, once again, it's in December, we have announced that we get 100% compliance of all the batches we have produced. The PDC lung, we call it PDC lung 01, the vaccine for the non-small cell lung cancer indications. So showing that we have the CMC completely under control today, and we hope that it will carry on like that. And as you know, in cell and gene therapy, CMC is really key. Okay, let me show you now how the technology works. So as I said in the beginning of my presentations, we have established a master cell bank with the cell line from the PDC leukemia patients that are being purified and amplified and put in bags in liquid and stored in liquid nitrogens. You see here the tank of liquid nitrogens. And from this master cell bank, we have developed several working cell banks. And when we want to launch a manufacturing campaign, we just have to take one bag of the working cell bank, seed a single-use bioreactor. We have 1 liter and 10 liters bioreactors. After 9 to 10 days of cultures, we have more than 20 billions of cells. The cells grow in suspensions in the bioreactor without any calf serum. It's a synthetic medium from Lonza. And at the harvest, we just have to split the cells in bag according to number of peptide we want to load. We load peptide, the GMP peptide solubilized in DMSO in the bags, and the peptide will stick to the HLA present in the membrane of the cells. Then we pull the different bags, irradiate the cells to stop the proliferations. Then the cells are put in a specific vial, the technology of aseptic technology with a cryoprotectant. And we, like for the COVID-19 vaccine, we just have to ship the cells in -80 degrees fridge. And when the patient is ready, we just have to thaw the cells and with a syringe, take it from the vial and infuse to the patient. So it's extremely easy and process. We have an off-the-shelf, so it's very scalable. And we have, in terms of batch-to-batch consistency and cost of good, very interesting data today. That is for the manufacturing. Now, in terms of potency, we did different benchmark study to demonstrate the capacity of the cell line to expand the CD8 T cells, priming and expansions of CD8 T cells specific of different antigens. And you see here an example by fax dextramer, it's in vitro, of the expansions with one specific antigen, which is melanin. And as you can see here with the cell line, we are 20 to 200 for more potent than the benchmark, which are the conventional autologous or allogenic myeloid DC. So showing once again the potency of our cell line to make the job, which is to expand CD8 T cells. We did as well study with neo antigens to show that the cell line can expand any kind of antigens. And here you see the two neoepitope, which are mutated peptide that we have identified in the literature. It was this peptide, the mutation in red has been clearly established, and they were identified in the blood of melanoma and lung cancer patients. And we have loaded the cell line with each peptide and put in present with purified CD8 T cells of 3 healthy donors. And we have measured after 2 weeks and 3 weeks of culture the expansions of CD8 T cells specific of each of these peptides. And as you can see here in blue and in red and purple, we have definitely been able to show a very nice expansions of the CD8 T cell with a range of 154 degrees. And this result was extremely interesting because in healthy donor, the precursor of this neoepitope CD8 T cell is extremely low. It's 1 out of 1 million, as all the precursor are naive. Okay, so today we present HLA-A2 restricted peptide. It's a selection criteria. We select in the current clinical trials HLA-A2 patients. But we have modified genetically the cell line to make it expressing additional HLA like the A24B35, and it works quite well, very well. We have published this data earlier this year, and with B35 restricted peptide, we can definitely expand B35 restricted CD8 T cells effectively and with a high range. So as I said, we are loading the cell line with GMP peptide today because it's the easiest approach, but we have as well tested other approach like retroviral transductions of polypeptide or full antigens, and to have endogenous presentations of the peptide by the cell line. It works very well. We did as well electroporations with mRNA of the cell line, and it works very well as well. So in terms of tumor antigen loading strategy, we have different pathway, and we can adapt according to the indications. In terms of IP, we have 3 key pillars in our IP strategy. First of it, we have the master cell bank, which is clinical grade, unique worldwide, and licensed from the French Blood Bank. We have the patent. 2 patents has been granted, and 1 on the genetic optimizations of the PDC line is ongoing, is in PCT process. And the third pillar is all the know-how and data we have generated related to the manufacturing process and QC, the immunomonitoring assays, and the preclinical data. Okay, let me show you now the result of the first academic clinical trial we did with melanoma. So the cell line were loaded with 4 different antigens, tyrosinase, GP100 melanase, and MAGE3, and we have treated 9 patients. We tested 3 doses, low dose, mid dose, and high dose. The vaccine has been infused weekly 3 times to each patient. The results were definitely promising. We have observed an acceptable tolerability and safety profile. That was the primary objective of the study. We didn't observe any allogenic rejections detected, meaning no antibody, mismatch antibody identified in the cell line in the blood of the patient, after the 3 infusions. And we have observed significant antigen-specific CD8 T-cell response directly detectable in the blood of patients. So let me show you an example of the data on 1 patient on the immuno result. So this is the CD8 T cells quantified by FACS dextramer. And as you can see here, week 0, so just before the infusions of the first vaccine, we have 0.17% of melanase CD8 T cells. That is the result for the melanase. After 1 week, we pass to 0.15. 9 weeks, we go to 0.44. 54, and after week 15 we go up to 0.86. So we have more than 5-fold increase in terms of the CD8 T cells after the 3 infusions. And what is extremely interesting to see as well is the profile and naive and memory T cells of these CD8 T cells, and for that we have been able to look by over marker like the CCR7 and CD40A, 45 array, according to the profile and expressions of these different proteins in the CD8 T cell. We can quantify the pool that are naive. You see here are the naive T cells, and below are the memory T cells. Here are the memory T cells. So you see, just before the infusion, we have more memory, 60% of memory and 41% of naive. And after the 5th weeks, the 3 infusions, we go to 86.2% of memory. So we have a switch from naive to memory thanks to the activity of the vaccine, which is what is expected with a cancer vaccine. And this result was very interesting to get because we just infused 3 times, and we know 3 times is not optimal. And you will see in the non-small cell lung cancer we are running today, we're infusing 6 times. And despite the fact we didn't co-administer any checkpoint inhibitor. So this result was really with the vaccine alone. And we know that very synergetic effect with checkpoint inhibitor. You see an example here, the result of an experiment we did with the blood of melanoma patients. You know, melanoma is a very hard tumor with a very high tumor-induced immunosuppressions. And we have loaded the cell line with melanase again and put in culture with the PBMC, the white blood of the melanoma patients, and look after 1 week and quantify the CD8 T cell expanded. You see here for the vaccine alone, we have 15-fold increase. With the checkpoint inhibitor alone, we have multiplied by 2, which is not statistically significant difference from the baseline. But when you combine both, the checkpoint and the vaccine, we have 35-fold increase, and it's where you observe this very nice synergetic effect. This data has been published, and you see the reference here last year in oncoimmunology, and it has been done in a cohort of 13 patients, melanoma patients, the blood of 13 melanoma patients. Okay, let's pass now to the result of the lung cancer trials, non-small cell lung cancer trials. So let's show you here the 6 different antigens we have selected for this indication. So we have MAGE4, MAGE3, MUC1, New York ASO1, Trevin, and Multimage. And as you can see here, for each of in vitro with the blood of healthy donor, we have been able to amplify the CD8 T cells for each of these antigens. And you can see the frequency is quite high for some of them. lower for others, but it's quite well known in immunology to have this kind of heterogeneity. But we have been able to show for each of these antigens that we can have significant expansions of the CD8 T cells. What you see here is the design of the trials, the clinical trials. So it's conducted on 64 patients. Objective is to assess safety, immunological activity, and clinical response of the vaccine with and without checkpoint inhibitor. So we test 2 doses with the vaccine alone in monotherapy. So the low dose, which is 2 million cells per peptide, and high dose, which is the 20 million cells per peptide. First in monotherapy in adjuvant settings, so it's just after surgery. It's phase 2B, phase 3B, and 3A patients, where you can do the surgery and after you have a adjuvant chemotherapy, and after a washout, we infuse 6 times the patient. So the infusions, the vaccine is infused 6 times through IV and subcutaneous. And after these 2 first cohorts, we have the 2 last cohorts, which are done in first line metastatic patients on naive to checkpoint inhibitor, but in association with checkpoint inhibitor, with low dose first 6 patients, and then we go to the high dose with checkpoint inhibitor on 42 patients. 42 is because it is in this last cohort we want to observe the clinical response. So we have already completed the 3 first cohorts, and we have already opened the last dose, and a few patients has been included. Okay, just to finish my presentations, just the timeline of the company. So we have started the clinical trial in 2020. So we expect to finalize the clinical to get the final clinical study report with the PFS 9 months of the last patients, end of 2023. And we get 20 million euros of funding end of beginning of last year, and we expect to raise a new round of financing this year to complete these clinical trials. And after these clinical trials, the objective will be to have a phase 2/3 randomized study to demonstrate the clinical benefit of our vaccine, and to get registrations and to work on new indication or to set up in US and Asia. Of course, we will carry on to work on the platform. I didn't have time to present you, but we know the mechanism of action of our cell line. We are working on how to even optimize the capacity of the cell line to work on new antigens or on the CMC development. I would like to thank you for your attention with a nice picture of part of the team based in Belgium and France.
Hi,
So
Eric.
Yes.
Can you hear me?
Yes, I can.
Okay, thank you so much for your very informative session. You have touched so many critical areas which is solid need of the hour, and I hope everyone here have enjoyed your presentation. And we are looking forward to work again with you. I'll ask the audience that if they have any questions, they can ask in the QA session or in the chat. Okay. Thank you so much, Eric. Stay safe.
Thank you.
Thank you for joining us.
Thank you. Have a great day. Bye.
You too. Stay safe. Okay, so ladies and gentlemen, this has been a wonderful conference. I have enjoyed listening to the talks from our valued keynote speakers. It is a privilege and honor to be entrusted with such an undertaking at a gathering of such eminent people from different parts of the world. So thank you so much to our esteemed presenters for their valuable point of view. Now we have reached to the end of our conference. On behalf of Platinum Global Solutions, I would like to thank all the stakeholders, including speakers, sponsors, and delegates, without whom this conference would not have been possible. I extend my best wishes to you all. We will be back with novel themes in the coming months in the field of drug discovery. And we are having